Pero ya están hablando.
Ahora empezamos.
Ya. A ver, veamos qué pasa.
Ya les diré algo porque no vinieron. Tenemos 5 líneas de investigación, ¿ya? Vamos a ver. Tenemos un total de 15 becarios ANID, ¿ya? Algo que para nosotros es súper importante, el poder que nuestros estudiantes accedan y puedan obtener becas ANID es súper importante para el programa, tanto para el propio crecimiento del programa, como también para que el programa tenga recursos, como por ejemplo para hacer esta importante actividad. No quiero aburrirlos con esto, solamente voy a tratar de pasarlo rápido. El objetivo de nuestro programa obviamente es formar capital humano avanzado de la más alta calidad, capacitados para desarrollar investigación básica y o translacional en las distintas áreas del conocimiento que se aplican a la biomedicina, con énfasis en las enfermedades humanas, y que contribuyan a la búsqueda, desarrollo y aplicación de nuevos conocimientos que aporten a la salud de la población. ¿Ya? Los objetivos específicos ya están ahí, los pueden ustedes mismos ver, pero somos un programa experimental, un programa de doctorado experimental, presencial, y que ahora, como les decía, está acreditado por 5 años por los CNA. Estamos acreditados hasta el 2026. Como programa de doctorado. Estos son el perfil de ingreso y los requisitos de admisión. Los muestro muy rápidamente para que sé que aquí hay estudiantes también de pregrado, para que se vayan motivando y se vayan interesando también en cómo hacer para estudiar los programas de posgrado. Aquí está la información que necesitan saber para los requisitos de admisión, el perfil de ingreso y los requisitos de admisión. Muy rápidamente el perfil del graduado, que no lo voy a comentar, pero que nuestro objetivo, como decía, es formar investigadores independientes que puedan contribuir con soluciones innovadoras a mejorar la salud de las personas. Finalmente, ahí están los requisitos de graduación. Nosotros exigimos varios aspectos para que un estudiante se pueda graduar del programa, y dentro de los más importantes es que tiene que tener al menos 2 publicaciones, una enviada y una ya aceptada, donde en una de ellas tiene que ser el autor principal. Ahí está mi información. Yo soy Rodrigo Mur Carrasco, soy el director del programa todavía. Ahí está mi correo electrónico. La convocatoria 2024 ya está cerrada, obviamente. Logramos aceptar para el año 2024 9 estudiantes que en este momento están compitiendo por las becas ANID. Si aquí hay becarios o estudiantes o postulantes que están esperando las becas, seguro que todavía están con muchos nervios, no han salido todavía los resultados, se supone que salen esta semana. o la próxima, entonces nos tienen ahí a todos muy nerviosos. Muy brevemente también sobre el Magíster en Ciencias Biomédicas, el programa también que está organizando este evento. El Magíster en Ciencias Biomédicas tiene una trayectoria mucho más larga que la del doctorado. El Magíster en Ciencias Biomédicas nació en el año 2005, tiene 22 años de trayectoria, ha graduado un gran número de nuevos magíster. Es uno de los programas más antiguos de la Universidad de Talca. Está actualmente acreditado también por la CNA por 5 años, hasta el 2026, igual que el programa de doctorado. Es un magíster científico académico de carácter presencial y de dedicación exclusiva, al igual que el doctorado. Tiene menciones ahora tiene 3 menciones, que son la de bioquímica e inmunohematología, la de microbiología clínica y la de patología oral. Ahí, bueno, está el perfil de ingreso, no voy a entrar en detalles de él, solamente decirles que son 2 programas que están acreditados, que son del más alto nivel y que los esperamos aquí en Chile, aquí en Talca, a los que quieran venir a formarse con nosotros, que quieran confiar en nuestro programa para que los formemos aquí en Talca, los estaremos esperando. Solamente, bueno, ahí los detalles de cómo postular al programa de Magíster. La convocatoria del año para el 2024 ya también está cerrada, pero igual les dejo invitados a conocer el programa, a que investiguen sobre él si les interesa hacer su formación de magíster o doctorado con nosotros. Me pueden contactar a mí o pueden contactar al doctor Sergio Bechinger, que es el director del programa de magíster. Acá el afiche de esta actividad que vamos a tener en el código QR que está al costado derecho de ustedes. El código QR se lo lleva a la transmisión por streaming de la actividad. Si ustedes tienen contacto, amigos, personas que quieran conectarse a través de streaming, pueden escanear ese código QR que los va a llevar directamente a la transmisión. También está ese link que está ahí arriba, que también los lleva a la transmisión online. Y bueno, me voy a tomar la libertad de estos últimos minutitos que me quedan acá. Llegó el momento de dar las gracias. Yo, como les decía ayer a los profes con los que compartíamos ayer una cena, esta es mi última actividad o mi último Summer School como director del programa. Tengo que dejar el cargo, ya corresponde que lo deje, otro colega lo va a tomar. Y por tanto, para mí por lo menos, llegó el momento de dar las gracias. Entonces, quiero partir dándole las gracias obviamente a las autoridades que confiaron en mí para poder tomar este cargo y llevar este programa ya por casi 9 años. Lo acreditamos en un tremendo trabajo. Agradezco a mis colegas que confiaron en mí, a los colegas que integran el comité académico del programa. Les quiero dar las gracias por haberme apoyado y haber trabajado juntos, haciendo un tremendo equipo que logró, entre otros hitos, la acreditación del programa por 5 años. En una primera instancia, así a la primera, 5 años, algo muy bueno para nosotros. ¿Ya? Quiero dar las gracias a una persona muy especial, probablemente los estudiantes que están aquí del programa la conocen, Carolina Molina. Ella es la asistente del programa. Ella le entrega más que solo trabajo a este programa, le entrega también, ya que vamos a hablar de corazón, le pone mucho cariñosito al programa. Hace que este programa funcione, es la que permite que lata el corazón del programa. Ella lleva todas las cosas y quiero darle el reconocimiento ahora públicamente. Y también por haberme ayudado y haber tenido la paciencia de trabajar conmigo por estos 9 años en los que los dos tuvimos que ir aprendiendo muchas cosas. Y finalmente darle las gracias a todos y cada uno de ustedes que está aquí, en particular y en especial a los profes invitados, al doctor Gerardo, que vino, al doctor Gerardo, al doctor Flavio, que vienen desde México, nos vienen a visitar, y darle las gracias a cada uno de los expositores que aceptó estar en esta actividad. También un reconocimiento y un agradecimiento especial al doctor Andrés Cuez, que siempre ha estado ayudándonos desde el comité académico para también poder organizar estas actividades. Desde este punto en adelante la actividad hace un switch al inglés. Yo voy a dejar al doctor Andrew Quest en el escenario para que comience con la presentación del centro y de su centro y el ACTIS, y posteriormente ya empiezan las conferencias. Me corresponde entonces a mí presentarles al aquí. Ya. Entonces, yo voy a dejar en el escenario al doctor Andrew Quest. El doctor Andrew Quest es profesor titular de la Facultad de Medicina de la Universidad de Chile. Es el director del Centro de Investigación en Ejercicio, Músculo y Cáncer. También es investigador principal del ACDIS. Es un investigador con una larguísima trayectoria a nivel tanto nacional como internacional. Ha sido miembro del comité académico de nuestro programa durante ya varios años y nos ha apoyado desde ese lugar. También ha contribuido a la formación de diversos estudiantes también de acá de nuestra universidad, tanto en el programa de Magíster como de Doctorado. Es un gran honor para nosotros tenerlo aquí y los dejo con él. Le damos un aplauso al doctor Andrew Quest.
Bueno, gracias Rodrigo por la introducción. Muy breve, 3 palabras en español, después voy a hacer el switch. Esta es una actividad que llevamos adelante ya varios años, que es como la 5ª edición, ¿cierto? Cada año vamos cambiando el tema. Y justamente nosotros como SEMS y ACTIS, de cierto modo, actuamos como sponsors en esta actividad, por lo tanto me voy a demorar unos 15 minutos en explicar un poco qué son estos centros. Y a nosotros como centro, también en forma personal, me interesa mucho fomentar interacciones con las regiones, proyectos colaborativos, y de hecho le voy a mencionar algún ejemplo que hemos llevado adelante en forma exitosa. So, said that, I will switch to English. And I will be talking now about these 2 centres. The centre called CEMSA, which initially stood for Centre for Studies in Molecular Mechanisms of the Cell, and now, as of 2016, Centre for Studies in Exercise, Metabolism and Cancer. Okay. And the Advanced Centre for Chronic Diseases. So, what were or still are these centres? CEMSA, in its initial version, this is basically a photo showing the participants in the centre roughly 2010, 2011, shortly before the centre closed, that part of the centre. So this was a group of about 200 people, so by Chilean standards and probably many standards, a fairly large group of researchers, particularly a lot of students. This centre then evolved into a number of things, as you can see here. Is there a pointer somewhere? Rodrigo? ¿Hay un puntero? No, okay. So, as you can see, the beginning there is the Centre for Molecular Studies of the Cell, that then evolved into 3 new initiatives. First of all, an initiative called Nemesis, a network for metabolic stress signaling. So there was a certain shift towards studies on the relevance of metabolic phenomena in the development of diseases, and this then in turn evolved into a now existing centre, the ACTIS, the Advanced Centre for Chronic Diseases, and the new version of SAMHSA, the Centre of Studies on Exercise Metabolism and Cancer, as of 2016. So what do these centres have in common? Essentially, ACTIS seeks to identify common elements between cardiovascular diseases and cancer, amongst other things, and the SEMS in its current version seeks to study the causes of diabetes and metabolic diseases, particularly in the context of cancer. And the studies in both of these centres go from looking at individual molecules to studies in cells, organs, individuals, and also society-based studies. So, the PIs of SEMSE, current version, are from right to left, Sergio Lavandero, Cecilia Hidalgo, Enrique Jaime, and myself. And just for reasons of time, I would just focus on telling a little bit about some of the studies we've conducted and we are still conducting, more related to my line of research. Okay, so as I said, I'm particularly interested in mechanisms that favor diabetes and also cancer, so metabolic phenomena. These studies on diabetes we did together with Sergio Wenger here in Talca, now director of the program, Magíster. Okay, so a protein we are particularly interested in, and you'll hear more about this in my talk tomorrow, is a protein called caveolin, and this protein has many roles, as again we'll hear more about, but it started off as being a protein implicated in cholesterol transport and also plays a structural role in cells, forming the co-protein of caveolin. Then it became clear that it plays an important role in the development of cancer, initially as a tumor suppressor, so this would be number 2, and then a role was identified for the protein in promoting migration, invasion, and metastasis, which obviously would seem to be a contradiction, and I'll try to resolve that contradiction tomorrow. And more recently has been implicated as a component of extracellular vesicles, in particular exosomes, and our current interest focus mainly on that aspect of caveolin. So we studied for many years what I call cell intrinsic mechanisms of signaling, and the mechanisms we study now in exosomes are what I refer to as cell extrinsic mechanisms. So here just an example. Well, this is an experiment from the beginning where we showed that actually caveolin acts as a tumor suppressor. So these are colon cancer cells in a xenograft model, nude mice. And as you can see in the lower panel to the right there, those cells that express caveolin don't form tumors or do so less efficiently. And one aspect of cancer development we've become particularly interested in is that of angiogenesis, which is absolutely essential for tumor growth. So this is what you see here. Tumors cannot grow beyond a very minimal size without angiogenesis. And I'll talk a little bit about that tomorrow too, but one project of interest here is a project we developed with, or we participate in with Roger Moore, and that has to do with vascular senescence. Okay, so now getting to the diabetes part of what we do, or have been working on. This is sort of human development over this part several millions of years, and this part only about 50 years. Rather a sad story, obviously not a very beneficial development. And together with Sergio Wenger, we've been working on how, for instance, our favorite protein, caveolin, participates in these events that lead to beta cell death, either using cells in vitro or also using models in vivo. So when caveolin is present and stress situations like exposure to palmitic acid or high-fat diet in a mouse, the presence of caveolin in these cells favors cell death and therefore elimination of these cells, and therefore development then eventually of type 2 diabetes and then type 1 diabetes. As I said, we've now focused a lot on research that has to do with exosomes. So what are exosomes? These vesicles are formed in a multivesicular body and then are liberated and then taken up by receptor cells or recipient cells and induce changes there. And so we're very interested in understanding how these changes in recipient cells are brought about. And more recently, we've developed together with Mariana Cifuente, who you'll hear about or hear from later on today, how also adipocytes here, they also produce vesicles. These vesicles can also impact on tumor development, something I'll talk about tomorrow. This is sort of the working hypothesis of one of the projects we have together with her. And the student Sophia Samwesen. So basically the idea is that these vesicles would impinge on macrophage polarization, and these macrophages would then somehow participate in the tumor formation process. A project we're developing again with Rodrigo Moore here in Talca is a project related to cachexia. This is what we see here. So the idea there is that tumors produce certain vesicles. These vesicles are liberated and induce a phenomena in muscle that leads to essentially loss, so muscle atrophy, as you can see here, by stimulating proteolysis, muscle cell apoptosis, and inhibiting myogenesis. And so this is a project that is just beginning with a joint project. So those are some of the scientific initiatives that are ongoing in SEMSE. Let me now just tell you a little bit about what else SEMSE does. So SEMSE is a facility, has several pieces of equipment that can be of interest, and a piece of equipment that anyone can use, not just people in the center, also people in other universities, also the University of Talca. And so we have a flow cytometer, we have a C2 confocal microscope in the center, and then several other types of microscopes, a spinning disc, another confocal here, an older model, and quite importantly now, an ultracentrifuge that allows us to isolate particularly extracellular vesicles and exosomes. We have a virus facility. This allows us to work with pathogens. So anybody who'd like to work with pathogens and would like to collaborate with us, we have the facilities to do that. And we also have an animal facility here in particular where we do work with mice, particularly the tumor xenograft experiments. So, as I said, I invite you to look at the website here. If you want to know more about services offered by SEMSE, and if you need any more information, contact those people there, Hervé and Cecilia, or just write me. Now to ACTIS. So this is a center, joint center between the Universidad Católica de Chile and the Universidad de Chile. And the idea is to define common causes for chronic diseases, so particularly cancer and cardiovascular diseases, and this in the perspective of defining now strategies to control disease development and progression. Here we have a timeline of the Advanced Center for Chronic Diseases. So essentially it was started in 2013, 10-year period, 2023. So essentially ACTIS in its initial projection has come to an end, so to say, but we have been awarded a continuation project now, so with this continuation project, ACTIS continues to function, 2024-2025, and in this period we are expected to come up with a new proposal for renovation of the centre, which could then be quite different from the existing centre. Anyway, these are the PIs in the centre, so Sergio Lavandero, our director, Paolo Gattaro, both work on cardiovascular issues, Alejandro Corvalán and myself on cancer, these days particularly gastric cancer. Caterina Verrecchio involved in cancer epidemiology, and Marcella Kogan, he is focused on nanomedicine. Then we have a core project called the Maule Cohort Project, which is a population-based study where we recruit people in Colina, and essentially 10,000 people have been recruited, and we have information available on all these people and also samples, and so different kinds of studies can be done with these samples. And then we have a basic sort of preclinical and clinical core facility. Now beyond these 6 people, we now have a new acquisition for over 2 years now. This is Mariana Cifuente, she's here in the back somewhere, and you'll be hearing more from her, and she is a PI in the centre now, and her area of expertise is obesity and basically adiposition, which is obviously a key issue we'll be dealing with today and tomorrow at this meeting. So the projects in ACTIS cover many areas, as you can see, require individual efforts by different groups, but also they require to be successful sort of translational or cooperation between the different groups, so we can actually deal with and resolve more complex questions. The centre now has about, this is a somewhat outdated slide, but probably about 260 integrants. The important point here is that most of these people in this centre are people in training, so either undergraduate students, master students, graduate students, or postdocs, 60% in total. Publications, I would think the centre has been rather successful in this respect, well, in many other respects too. So over 670 publications now in about something like 10 years, average impact factor over 6, 430 in Q1 journals, 120 in top 10 journals, and interestingly also 16 patents. So there is definitely an effort here to generate information, knowledge, that can then somehow translate into, for instance, products. I'll give you an example of that in a minute. Then we have an outreach program that has been overwhelmingly successful. This is just a summary of the information available for 2022. So 400 events were organized in which there were over 22,000 participants. And so there are all of the activities that we developed on the city. So beyond that, the centre received international recognition by the CIMAGO institutions ranking, CIR, as you can see here for the years 2020, 21 and 22. It was ranked third, so the third most important institution in scientific investigation in Chile, just behind the Universidad de Chile and the Pontificia Universidad de Chile. So this is quite an amazing achievement, considering that we're talking here about a centre with 260 integrants, and it's being compared with universities that have, of course, several thousand people that work in these universities. Obviously not only doing research, also doing teaching. Just briefly again, the major areas of interest. So digestive cancers at this point might change in the future. Heart failure, and then these transversal elements like nanomedicine, epidemiology. And so how do we generate these connections between individual projects? We do that with these using students who are co-mentored by 2 or even 3 mentors belonging to different research lines. We of course MAUCO, which was inherently permits connecting research lines. And there's also internal funding, as you can see here, for generating new sort of transversal projects between research. So just a few examples of research in the centre. This is just an example from our group, working with other groups, on the role of Helicobacter pylori in gastric cancer, so this is well documented. And we became interested in what are referred to as outer membrane vesicles. these things here. They're produced by bacteria, so much like cells, cancer cells, and other cells. Bacteria also produce vesicles. They're actually quite similar in size, not necessarily in composition, obviously. There was a considerable amount of information available, epidemiological information suggesting that Helicobacter pylori infection may not only lead to or cause favour the development of gastric cancer, but may also be relevant in the development of systemic diseases and things like Alzheimer's, for instance. And so we focused on this aspect in more recent studies, and were able to show that vesicles, these here, can actually get out of the stomach, produced by the bacteria, can get out of the stomach, can get to the brain, and can induce damage in the brain. So this is sort of summarized here. So these vesicles get to the brain and actually damage neurons there. It's probably a direct mechanism, there's also an indirect mechanism. And so this was published in this journal here, which is quite prestigious. This is an example from work done in Marcelo Kogan's group, where they're looking at different types of nanoparticles and how they can be modified and then used to carry drugs to specific sites in the body. This is an example, again, a collaboration between Marcelo and our group, where we used nanoformulations of curcumin to treat, obviously, cancer in mice, both sort of subcutaneous tumor formation and also metastasis was actually very successful, led to quite an important publication here. And so this is sort of an example illustrating how the collaboration between quite different research lines can lead to quite interesting results. Then we have here another one between Marcelo's group and our group, where we used exosomes to target nanogold, so gold nanoparticles, actually to specific sites in the lung. And the idea there is to carry drugs to these specific sites, and then you can irradiate with infrared light and basically liberate the molecules from the surface of the gold particles at specific sites in the body. And so that means you can actually do targeted delivery of otherwise highly cytotoxic molecules, which when administered systemically has severe side effects. And this is just the last example, one again with the nanoemulsions, the curcumin nanoemulsions, and we found that these are actually quite useful in killing Helicobacter pylori. So one of the things we're working on now is showing that we can actually use these kinds of formulations to treat people. So we're initiating studies with people, and the idea is that we could use these formulations to eliminate the bacteria, preferentially in children, because they're the ones that get infected rather early on and have lifetime experience with bacteria. And it would be desirable to use things other than antibiotics, because antibiotic resistance is becoming a serious problem worldwide, and of course also in China. And just to finish up here, I would like to mention that this nanoformulation, nanocurcumin, is now also available on the market as a product called NanoFix. So you can actually buy this. There is a company, Magíster, that produces this and then sells it, and so you can go into the website and find out more about that. So this is just an example of how we've gone from very basic research, which allowed us to set up these animal models that allowed us then to test the efficacy of certain combinations of drugs, and then, for instance, these nanoformulations, generate such formulations for scientific purposes, and then also from then on going on to generate a product that you can actually buy on the market. So that's actually quite an amazing transition, I think, and obviously something highly desirable. Malco, of course, the cohort I already mentioned, 10,000 people already recruited. Information available on these 10,000 people and ready for collaborations of many different kinds with all sorts of groups. Whoever's interested can just contact Katerina for 88. and try and come to an agreement on what you want to do. So I think it's safe to say that ACTIS has consolidated itself as an important transdisciplinary research centre in Chile. Of course, we hope will continue existing beyond 2025. So that's it for today. Thanks for your attention, and if anybody has any questions, happy to take. That's it for today. Thanks for your attention.
Well, continue with the program. Good morning, everyone. I'm Professor Daniel Gonzalez, part of the steering committee of our PhD program in biomedical science. I have the pleasure to present our first speaker, who is Professor Gerardo Garcia Reyes. He belongs to the Tecnológico de Monterrey in Mexico. Professor Garcia Reyes did his undergraduate and PhD studies at UNAM, the Universidad Nacional Autónoma de México, and then he did a postdoctoral position, research position in University of Wisconsin, Madison, in the field of cardiac electrophysiology, and then he moved back to Mexico when he incorporated to Tecnológico de Monterrey, where he belongs to the School of Medicine and Health Science and the Institute for Obesity Research. Has appointment in both entities. In the School of Medicine, he's a lecturer in cardiology. He's professor of cardiology, and he has several recognitions. He's part of similar our ANID, CONACYT in Mexico. It's similar our ANID. He's a member of the study groups there. He's belonged also to the Academy of Science in Mexico, and he's director of experimental medicine and leader for the director of the National Director for Translational Research in this Institute of Obesity Research and Instituto Tecnológico de Monterrey. So let's give a warm welcome to Professor Gerardo Rivas. Today he's going to talk about his field of expertise, that is mitochondrial dysfunction in heart failure. So, Professor, welcome. Thanks for coming. This is his second time in our university, at our university. So, thanks again for coming here, and we will follow your talk. Thank you very much. Let's give him an applause.
Muchas gracias por la invitación, particularmente profesor Rodrigo, que nos ha hecho el honor de invitarnos, y sobre todo estoy muy contento también de ver la evolución que ha tenido el programa de posgrado. Justamente comentaba con el profesor Daniel que hace unos 6 años estuvimos por aquí, y ver la evolución del programa, cómo ha crecido, cómo ha sido certificado, pues es algo que nos llena de muchísimo gusto. Voy a empezar un poquito platicándoles sobre la institución. Creo que un poquito el foco Institute for Obesity Research. Perdón si hago cambios en inglés y español, pero traigo en la presentación en español. Ayer traduje todo pensando que era en castellano, pero puedo hacer switches. Lo que quisiera comentarles inicialmente es que el Tecnológico de Monterrey es una universidad preferentemente docente. Ha tenido muy buenos programas educativos y con ello ha logrado una reputación académica en la región, y actualmente, hace unos 20 años, tomó la decisión de moverse a temas mucho más de investigación, y particularmente en salud hemos tomado la decisión de hacer inversiones en áreas muy estratégicas, y los 4 centros que aquí les presento es donde en salud hemos decidido invertir nuestros recursos, nuestro capital humano particularmente, y aquí les platico los 4 proyectos centrales más importantes. Sin dar muchos detalles, quisiera mostrar un panorama muy general. El primer programa se llama Origen, que es un programa de secuenciación de exoma y un biobanco que recluta 100.000 mexicanos para poder tener datos y muestras de los mismos, aprovechando la infraestructura del Tecnológico de Monterrey, que tenemos 29 campus en todo el país. El siguiente centro, que es donde participo y tengo el honor de coordinar una unidad, es el Instituto de Investigación en Obesidad. Un tercer centro es un centro de excelencia en falla cardíaca, y el cuarto centro importante en investigación en salud es el Centro de Cáncer de Mama, Tumor Center, que particularmente está enfocado en el estudio de cáncer de mama en mujeres menores de 40 años. Profundizando un poquito en el Instituto de Investigación en Obesidad, quiero comentarles que está compaginado o integrado por 5 unidades de investigación. Aquí ven los 5 grupos. Es un instituto multidisciplinario en donde tenemos temas de alimentos saludables, biología integrativa que recoge todos los datos de ómicas, genómica y el proyecto Origen, un área de dispositivos biomédicos, y también tiene interesantemente un área de política y emprendimiento público. A un servidor le toca la responsabilidad de coordinar el área de medicina experimental y terapias avanzadas. Y nuestro modelo es muy sencillo, es un modelo muy clásico en donde empezamos con investigación e identificación de potenciales compuestos, fármacos que tengan algún potencial, el cribado en sistemas de rápida escala, posteriormente escalando a sistemas de tercera dimensión, en células pluripotentes que generamos variantes propiamente de la población mexicana en donde probamos las drogas de eficacia, y por supuesto la expertise que se tiene en la generación de pruebas de concepto en modelos preclínicos. Y con todo ello poder llegar justamente, como lo profesaba el profesor, justamente la posibilidad de trasladar todos esos hallazgos hacia una población. Este es nuestro centro y estos son la gente que está incorporada. Ahora nos acompaña también de nuestro equipo el doctor Pedro Contreras, en donde estamos divididos en diferentes áreas, desde el cribado in silico hasta las pruebas in vitro, la formulación y posteriormente pues equipo también clínico que se ha encargado de trasladar los hallazgos. En ese sentido, muy generalmente es lo que estamos haciendo en nuestra unidad de medicina experimental. Y como bien dice el profesor Daniel, ahora voy a platicarles un poco del área de expertise en donde hemos trabajado por mucho tiempo, que tiene que ver con la insuficiencia cardíaca y la disfunción mitocondrial. ¿Por qué venir a una conferencia de obesidad platicándoles de insuficiencia cardíaca? Porque las poblaciones en Chile, igual que en México, están sufriendo una transición demográfica muy relevante. Aquí podemos ver, son datos puntualmente de México, en donde vemos que en los años 50 la población, particularmente la pirámide poblacional, estaba particularmente con gente muy joven. Ahora podemos ver los estimados para el 2050, que es muy similar a lo que ocurre también en Chile, en donde todos los individuos ahora estamos mucho más, la pirámide está movida hacia mayores de 40 a 60 años, y entonces esto está claramente asociado con un incremento del riesgo cardiovascular y la enfermedad metabólica. Además de que tenemos más años debido a esa transición demográfica, también las poblaciones, entiendo, desafortunadamente también en Chile como en México, somos poblaciones que tenemos más kilos. Y esta prevalencia de sobrepeso y obesidad en México, siendo en Latinoamérica la población más alta, muestra que ya en 2018 en individuos adultos de más de 20 años tenemos más del 76% de individuos con obesidad y sobrepeso en nuestro país. Aquí vienen separados por obesidad y sobrepeso, en lo cual incluso con este triste dato en población pediátrica podemos observar que ya el sobrepeso y la obesidad en niños de 18 a 19 años llega a ser en niñas hasta el 27-30% de la población. Tenemos un terrible... Y esta asociación clara que existe entre más años y más kilos, pues nos lleva a problemas cardiovasculares, y particularmente yo les voy a hablar de la falla cardíaca. La falla cardíaca es un síndrome complejo en donde ya la capacidad de bomba del corazón no permite cumplir las demandas metabólicas del cuerpo. Sin embargo, un tema muy relevante asociado con obesidad es que la falla cardíaca, tal como la conocíamos, ha transitado de ser una enfermedad de bomba, observada principalmente en individuos fumadores, hipertensos, después de una enfermedad coronaria, a una enfermedad que tiene un contexto particularmente metabólico. La enfermedad que conocemos como falla cardíaca, heart failure with reduced ejection fraction, que tiene un gran arsenal de moléculas para tratarse, ha evolucionado a una nueva forma que conocemos como la heart failure with preserved ejection fraction, o la falla cardíaca con un trastorno de disfunción diastólica. Y algo que quiero mencionar claramente es que aquí está muy asociada, particularmente en mujeres que presentan obesidad, hiperlipidemia, diabetes está importantemente asociado, Y lo más relevante hasta el momento, no tenemos terapia para poder tratar a estos pacientes. Mientras en los años 80, 20% de la población con falla cardíaca presentaba esta forma metabólica de insuficiencia cardíaca, ahora los estimados son hablar del 40% y se espera para el 2050 que sea la forma más predominante de insuficiencia cardíaca sin opciones terapéuticas. Con esta justificación, pues hemos tratado de pensar nuevas posibilidades, salir de los marcos de referencia para plantear el desarrollo de nuevos fármacos que potencialmente puedan ser útiles en una enfermedad que contiene una base metabólica. Y con ello voy a meterme unos cuantos minutos a describirles la relevancia que tiene el organelo productor de energía en las células. Aquí podemos observar en forma general una célula cardíaca, estos miocitos de los que hablaban hace un rato, que tiene una estructura muy específica y en donde sabemos que hay algunos elementos muy relevantes que sugieren un profundo impacto de la función mitocondrial en las células cardíacas. Particularmente quisiera mencionar que la energía producida produce más del 95% a partir de la fosforilación oxidativa. O sea, la mitocondria en el corazón es el responsable mayoritario de la energía necesaria para la contractilidad. Adicionalmente, si tomáramos la cantidad de proteína de origen mitocondrial, más del 40% de la proteína en un miocito es específicamente mitocondria. Adicional a ello, tenemos que hay diferentes poblaciones especializadas en las células cardíacas, y me gustaría mencionar que más allá de un centro productor de energía, la mitocondria se ha convertido en los años recientes en un regulador del balance redox. Sabemos, es crucial en la muerte celular por diferentes vías, y también sabemos que contribuye al manejo de calcio intracelular, señalización celular. ¿Qué tiene que ver esta mitocondria con eventualmente el desarrollo de falla cardíaca? Yo quisiera mencionarles que es un ciclo vicioso que se puede iniciar justamente cuando el corazón pierde, a partir del daño mitocondrial, un incremento de la glucosa anaerobia y disminución de la masa mitocondrial, la capacidad de producir energía suficiente, acumulación de lactato, lípidos, incremento del estrés oxidante, y todo esto lleva al principal mecanismo de disfunción cardíaca, que es la pérdida justamente de la reserva contráctil. Todo ello va perpetuándose en un ciclo en donde estos mismos mecanismos asociados con disfunción mitocondrial o estrés oxidante llevan a los cambios ya irreversibles de la insuficiencia cardíaca, que tienen que ver con una remodelación del tejido cardíaco y con claramente daños y síntomas de falla cardíaca. Un dato que me gusta mucho enseñar es un dato relativamente antiguo, pero muy marcador del papel que tiene la mitocondria en falla cardíaca. Es este resultado en pacientes en donde se estratifican pacientes que tienen un cociente, aquí medido como el cociente de la fosfocreatina versus ATP, o sea, buena salud mitocondrial, buena producción de energía, y siguen estos pacientes por años, por algunos meses, y observan la mortalidad. Los pacientes que vemos en línea punteada son pacientes que tienen una disfunción mitocondrial o disfunción energética en el miocardio medida por resonancia. Y podemos observar claramente que la mortalidad es mucho más alta en aquellos pacientes que tienen esta disfunción mitocondrial versus los pacientes que mantienen la función mitocondrial, lo cual nos hace pensar justamente que la mitocondria es un sitio importante de control de la función cardíaca y particularmente un blanco potencialmente terapéutico. Voy a regresar a una diapositiva de hace mucho tiempo, los cursos de bioquímica, para explicar cómo se produce toda esta energía de forma muy sucinta, espero. Recordemos que la energía en la mitocondria se produce a partir de la oxidación de nutrientes que generan eventualmente un gradiente electroquímico que permite justamente acoplar la síntesis de ATP a través del complejo de la ATP sintasa. Esa energía sale de la mitocondria y es llevada a los sitios justamente de utilización. El mecanismo que se conoce muy importantemente para regular el metabolismo mitocondrial depende de un incremento de la concentración de calcio en la matriz mitocondrial que conlleva a la estimulación de deshidrogenasas del ciclo de Krebs para incrementar la producción de equivalentes reductores. Por un lado, es demasiado importante estimular el ingreso de calcio en la mitocondria, pero si este umbral se supera, se lleva a cabo una sobrecarga que conlleva a un evento completamente opuesto a la estimulación del metabolismo energético, la producción de especies reactivas y la pérdida de la permeabilidad selectiva de la permeabilidad mitocondrial. Durante muchos años se sabían estos fenómenos, pero no se sabía quién era el responsable del ingreso de calcio a la mitocondria. Recientemente se identificó este canal iónico en la membrana interna, que se conoce, y es un poco el centro de nuestra charla, que se conoce como MCU por sus siglas de Mitochondrial Calcium Uniporter, que se sabe está en la membrana interna de la mitocondria y es la vía de ingreso de calcio hacia el interior de la matriz para los dos fenómenos, para la estimulación del metabolismo en condiciones fisiológicas y también para la sobrecarga de calcio en condiciones fisiopatológicas. Este transportador, que ahora sabemos es un canal iónico, está compuesto por múltiples unidades, tiene múltiples mecanismos de regulación, de los cuales hablaremos un poco y que son motivo de estudio en mi laboratorio. La primera aproximación, hace muchos años que iniciamos para poder estudiar el papel que tendría este canal iónico, es una aproximación farmacológica. Sabemos que este compuesto, llamado rutenio 360, que es un complejo nuclear de rutenio, es capaz de unirse con alta afinidad a esta parte del poro acuoso del canal y con ello bloquear su actividad. Entonces, muchos de los hallazgos que se generaron durante mucho tiempo son aproximaciones farmacológicas bloqueando el ingreso de calcio hacia la matriz mitocondrial. Nosotros hemos generando una cantidad de información importante en donde en modelos de infarto experimental, donde hay sobrecarga de calcio y consecuentemente muerte celular, a través de bloquear este canal hemos visto la protección durante la isquemia y la reperfusión, hemos visto que se disminuyen las arritmias, hemos visto, por ejemplo, este es un dato un poco viejo, pero de los primeros hallazgos, en donde en corazones de animales sanos se les sometía a un modelo de media hora de isquemia experimental y podemos observar que el desempeño mecánico de estos animales, controles, siempre disminuía después del proceso de infarto durante 30 minutos, cuando estaban protegidos con esta molécula que evitaba el ingreso al interior de la mitocondria, había una recuperación pues, muy cercana al control, e identificamos cuando medíamos la concentración de calcio en la mitocondria que cuando teníamos el inhibidor no se alcanzaba el umbral y con ello probablemente no se alcanzaban los mecanismos irreversibles de daño. Esto lo propusimos a partir de explicaciones farmacológicas, sin embargo, años después, cuando se identifica la molécula transportadora o el gen que codificaba este transportador, se recapitulan los datos observando que el infarto experimental en animales deficientes de esta proteína se reduce prácticamente a la mitad, sugiriendo que, como lo habíamos visto farmacológicamente, la sobrecarga de calcio de la mitocondria es un evento crucial asociado a la muerte celular. Hemos seguido explorando esto, recientemente observando en arritmias cardíacas. Esta es una arritmia asociada a sobrecarga de catecolaminas y como pueden ver en animales controles, cuando tenemos estas catecolaminas empiezan a aparecer fenómenos proarritmogénicos y termina finalmente en una taquicardia ventricular o muerte de estos ratones. Sin embargo, si se tratan estos animales de forma dosis creciente con esta molécula, la incidencia de taquicardia ventricular disminuye muy significativamente. La muerte celular en estas células, un poquito ya pasando a la era no de fármacos, sino a la era molecular de este transportador, cuando silenciábamos con RNA interferente la expresión de este transportador y sometíamos un periodo después de una hipoxia, podemos observar que claramente la apoptosis está reducida en células de animales que tienen baja la expresión de este transportador, nuevamente sugiriendo que la disminución en la expresión de este transportador conlleva protección en daño cardiovascular. Otros ejemplos importantes, por ejemplo, identificando ya la área molecular del transportador en un modelo de hipotiroidismo, donde sabemos que las ratas hipotiroideas están protegidas del infarto experimental, pudimos observar que el transportador propiamente cambia su expresión durante el hipotiroidismo e incluso proteínas asociadas al transportador cambian su expresión y con ello modifican la afinidad del transportador y por ello se piensa que estos cambios en la estequiometría de ese transportador contribuirían a la cardioprotección que se observa a eventos cardiovasculares de los pacientes hipotiroideos. Todo esto habla de un efecto de sobrecarga, cuando hay un exceso de calcio en la mitocondria y el bloquear el transportador puede tener efectos benéficos. Sin embargo, nos preguntamos en las condiciones fisiológicas en donde pues necesariamente tiene que haber un incremento de la frecuencia cardíaca como en una respuesta inotrópica, pues nos pusimos a pensar qué pasaría si es indispensable que cuando se incrementan las concentraciones de calcio en la célula cardíaca también deberían consecuentemente incrementar dentro de la mitocondria. ¿Qué pasaría si bloqueamos la respuesta del transporte? Y lo que observamos es que cuando nuevamente poníamos rutenio 360 para bloquear esta respuesta y veamos una disminución de la cantidad de calcio dentro de la mitocondria, la respuesta beta-adrenérgica está parcialmente bloqueada. Entonces, tanto tiene efectos benéficos protegiendo cuando hay sobrecarga, como tiene efectos deleterios que no permiten acoplar, digamos, la producción o la estimulación del trabajo cardíaco con las condiciones fisiológicas. Este hallazgo nos llevó a pensar en la hipótesis que les quiero compartir, que es justamente resumida en esta diapositiva, en la cual en condiciones basales el calcio tiene que ingresar a través del transportador, estimular el metabolismo energético. Cuando están catecolaminas o se requiere que el corazón trabaje a una mayor velocidad y a mayor fuerza, se incrementa la liberación de calcio que el transportador incrementa y con ello puede cumplir con las demandas. Sin embargo, en condiciones en donde esta estimulación de catecolaminas es crónica, está ocurriendo de forma constante como en falla cardíaca, y hay un exceso de calcio que está incrementando, el ingreso a través del transportador ahora puede generar probablemente efectos deleterios. Y sobre esta hipótesis es justamente lo que les quiero compartir un poco. Todos los experimentos previos fueron en condiciones agudas, en donde bloqueamos una arritmia o un infarto experimental. Sin embargo, transitamos hacia explorar qué papel tenía este transportador en condiciones crónicas. Y para ello nos ayudamos de modelos experimentales. Nuestro modelo más clásico de falla cardíaca es un modelo en el cual inducimos en ratones machos con un bloqueador de óxido nítrico sintasa, el ename, cloruro de sodio y posteriormente una bomba osmótica con angiotensina. Generamos una hipertrofia y una hipertensión en estos animales y lo comparamos con animales controles. En estas condiciones, aquí pueden ver, es un corte del ventrículo izquierdo del ratón, donde un control vemos claramente comparado contra un ventrículo con falla cardíaca, donde está dilatado, donde está hipertrófico, donde hay remodelación patológica. Todas estas fibras azules significan fibras de colágeno, pueden ver el tamaño enorme de los cardiomiocitos, y en estas condiciones observamos que en falla cardíaca había significativamente un incremento de la expresión de este transportador, y que también era concomitante con un incremento en la concentración de calcio mitocondrial, lo cual nos decía que en condiciones crónicas en falla cardíaca hay más transportador de calcio y también mayor concentración de calcio en la mitocondria. Había una correlación muy clara entre la hipertrofia que ocurre en el corazón en modelos animales contra la expresión del uniportador. En ese sentido, pues todo habla de una asociación, no vemos una causa a efecto, por lo tanto decidimos explorar con experimentos de silenciamiento en donde a células les disminuyamos la expresión de este transportador. Aquí vemos las células transfectadas y aquí las células en un modelo en el que podemos tratar de generar esa hipertrofia patológica. Lo hacemos con una concentración de angiotensina 2 y ustedes pueden observar claramente que la dinámica, igual que en el corazón del ratón, se observa cuando hay hipertrofia, la expresión de este transportador se incrementa. Obviamente cuando está silenciada la expresión del transportador, no es capaz de responder. El hallazgo más importante, y lo pueden ver en esta parte de la figura, es que cuando medíamos la hipertrofia de las células cardíacas, podemos observar en las células controles, cuando damos un tratamiento con angiotensina, las células prácticamente duplican su tamaño, lo cual implica hipertrofia patológica. Sin embargo, si ese transportador estaba silenciado, no observamos el fenómeno de hipertrofia. Y no solamente no observamos el fenómeno de hipertrofia, sino el resto de marcadores asociados con disfunción y remodelación patológica, como puede ser, aquí pueden ver la hipertrofia, pero pueden ver los niveles de péptidos natriuréticos que son marcadores de estrés cardíaco, el incremento en colágena, TGF-beta e incluso marcadores de inflamación que son activados por la angiotensina, pero en las células que tienen silenciado ese transportador y no es capaz de llegar a cabo la sobrecarga, están disminuido el efecto. No solo en esas células de mioblasto observamos este fenómeno, sino también en células de cardiomiocitos, en donde también observamos exactamente el mismo fenómeno. Cuando tenemos angiotensina, los cardiomiocitos hipertrofian, son neonatos, y cuando está silenciado el transportador, la angiotensina es incapaz de producir los cambios en remodelación patológica ni la tubulación distinta. ¿Esto a qué está asociado? Pues claramente a una disminución de la concentración de calcio dentro de la mitocondria. Ustedes pueden ver que con angiotensina hay un incremento notable en la concentración de calcio en la mitocondria y al estar silenciado el transportador no fue capaz de activarse. Este cambio en la concentración de calcio en la mitocondria está claramente asociado con un incremento de las especies reactivas de origen mitocondrial, con un cambio importante en este marcador de biogénesis mitocondrial y probablemente el efecto responsivo tenga que ver con este factor NF-κB, que sabemos que es reactivo a las especies reactivas mitocondriales. Podemos observar que cuando está angiotensina, la fosforilación de este factor, que está asociado con hipertrofia y remodelación patológica, incrementa su fosforilación, por lo tanto se activa y prende un programa prohipertrófico. Sin embargo, si tenemos bloqueado a la sobrecarga de calcio las especies reactivas de oxígeno mitocondriales, este factor no logra activarse. con ello no se prende el programa patológico. Eso es algo bastante interesante. Les voy a dar un resultado interesante en este momento. En estas células que están silenciadas del uniportador, como lo mencionábamos, tenemos fármacos para bloquear el ingreso a través del uniportador de calcio mitocondrial con el rutenio 360, pero también quiero mencionarles que la liberación de calcio o la salida de calcio de la mitocondria al espacio citosólico ocurre a través con un intercambio con sodio. Y este intercambiador sodio-calcio está modulado por este fármaco que se conoce como CGP. Aquí pueden observar que en condiciones, como habíamos mencionado, donde hay silenciamiento y las células no pueden hipertrofiarse en condiciones de angiotensina, aquí en azul, cuando nosotros químicamente bloqueamos la salida provocando sobrecarga de calcio en la mitocondria, ahora sí aparece el fenómeno de hipertrofia junto con el incremento del calcio en la mitocondria, la capacidad de producir especies reactivas de oxígeno y los programas prohipertróficos. Entonces, la mitocondria no es realmente el transportador, sino realmente la sobrecarga de calcio mitocondrial, el fenómeno que está disparando esta activación de fenómenos, de hipertrofia y remodelación patológica. Probamos también, por supuesto, con nuestra molécula estrella que hemos trabajado por muchos años, sintetizamos en nuestro laboratorio. Pudimos observar que el tratamiento en células controles tratadas con angiotensina, con el rutenio 360 farmacológicamente, somos capaces de reducir la hipertrofia, la sobrecarga y los mecanismos de remodelación patológica en una forma crónica. Lo pudimos recapitular nuevamente en cardiomiocitos neonatos, en donde también claramente vemos que el rutenio 360 reduce esta remodelación patológica. Y esos datos son interesantes, muy parecidos a datos recientes también del profesor Sergio Lavanderos, en donde ha identificado que fenómenos prohipertróficos como IGF-1, el rutenio 360 es capaz justamente de bloquear respuestas mitocondriales asociadas a hipertrofia a través de una vía, digamos, fisiológica con IGF-1. La vía por la cual decidimos explorar cómo es que la expresión de este uniportador se presenta sobre insuficiencia cardíaca fue a través de un estudio de curso temporal en donde explorábamos principalmente los dos factores transcripcionales que se han estudiado hasta el momento que regulan la expresión del MCU, porque recuerden que la hipertrofia incrementaba la expresión. Y entonces empezamos a tratar células en basal 3, 6, 12, 24 y 48 horas después del tratamiento con angiotensina. Seguimos el factor CRE, su estado fosforilado, y podemos observar la dinámica. Primero el sistema de la fosforilación de la sinasa de calcio-calmodulina. Podemos observar que es una respuesta casi inmediata al tratamiento con angiotensina y posteriormente disminuye. El factor CRE incrementa su pico máximo a las 12 horas, mientras que la expresión del uniportador es un poco más tardía. Entonces pudimos identificar de cierto modo que estas dos respuestas pueden estar involucradas en la expresión del transportador. Cuando hicimos experimentos para bloquear la respuesta de estos factores, tanto quitando el calcio con BAPTA, un bloqueador específico de la sinasa calcio-calmodulina NK93 o un factor de CRE, pudimos observar claramente que la respuesta de expresión depende de las dos vías, tanto de la activación mediada por calcio, debida por la sinasa dependiente de calcio-calmodulina, porque claramente bloqueamos la respuesta de expresión o sobreexpresión del uniportador, y también está asociada claramente con la señalización de la hipertrofia. Entonces, estos dos factores contribuyen al cambio en la expresión del transportador. Hasta aquí, pues todo sugiere que in vitro y en un modelo experimental podría funcionar la inhibición de este uniportador para evitar, o está asociada al menos, correlaciona con efectos benéficos de la remodelación patológica. Y pues en esta visión más translacional pues tenemos que ver que no solo es un fenómeno que encontramos en los ratones o en las células del laboratorio. Afortunadamente en mi laboratorio tenemos un banco de tejido cardíaco de seres humanos con falla cardíaca. Aquí les muestro una inmunohistoquímica de tejido sano, ventrículo izquierdo, de seres humanos sanos contra la remodelación patológica, y lo primero que identificamos fue un incremento en la inmunohistoquímica. Aquí tenemos cada uno de estos círculos representa a un paciente. Tenemos una corte de 25 pacientes con insuficiencia cardíaca de estado terminal, en donde podemos observar en línea punteada son los individuos control, y cada uno representa a los pacientes con insuficiencia cardíaca, donde podemos ver que incluso hay pacientes que tienen una expresión a nivel transcrito de más de 25 veces de este transportador. Entonces el fenómeno de sobreexpresión de ese transportador también ocurre en corazón humano de acuerdo a nuestros resultados. Los datos funcionales de estos pacientes, particularmente la fracción de eyección, que es un dato clínico muy relevante para estratificar el paciente en insuficiencia cardíaca, muestra que entre más uniportador existen en sus pacientes, la fracción de eyección está más comprometida. Y también el otro dato, estos son los diámetros en sístole y en diástole del ventrículo, lo cual habla de remodelación patológica. Nos indican que entre más remodelado o más hipertrofia se presenta, hay también mayor expresión del uniportador. Un dato que fue muy interesante es identificar en un mismo paciente qué pasa durante la transición de la enfermedad. El paciente, cuando está muy grave, en algunas posibilidades recibe tratamiento a través de un sistema de soporte ventricular. El ventrículo, a través de un sistema mecánico, deja de funcionar y la bomba extracorpórea se encarga de mantener la función cardíaca. Por lo tanto, el ventrículo deja de estar estresado y todos los factores que lo estresan y remodelan claramente se ven disminuidos. Tuvimos la suerte de tener estos 6 pacientes en donde en su estado de más disfunción pudimos tener una biopsia de su corazón y posteriormente al trasplante, meses después, donde ya el corazón está sin tanto estrés porque tiene una bomba extracorpórea, pudimos observar que la dinámica en condiciones más sanas, por así decirlo, del tejido cardíaco, hay una reducción de la expresión de este transportador. Entonces representa tener una dinámica y por lo tanto potencialmente podría ser un blanco terapéutico. En términos generales, hasta aquí el mecanismo propuesto es que la angiotensina activa la movilidad de calcio intracelular. Esto justamente activa las sinasas que permiten la sobreexpresión de ese transportador a nivel transcripcional. El incremento en ese transportador permite mayor ingreso de calcio a la mitocondria. Esta sobrecarga permite las especies reactivas de oxígeno que contribuyen a la activación de este factor NF-κB, que contribuye con la inflamación y el remodelamiento cardíaco. Esa es un poco la vía de señalización que identificamos. Por supuesto, se nota interesante cuando estamos en una condición de traslación, teniendo datos en ratones, teniendo datos en humanos, poder transitar a ver si tiene efectos terapéuticos y con ello, pues posicionar a rutenio 360, una molécula metálica que no ha sido probada en humanos, no es necesariamente la mejor idea. Entonces empezamos a buscar múltiples drogas y fármacos que pudieran tener eventos sobre ese transportador y nos encontramos particularmente con esta molécula, el cannabidiol, este activo no psicotrópico de cannabis, y sabíamos que tiene efectos directamente sobre el transportador, no en sistemas cardiovasculares reportados, pero sí en sistemas de cultivos de neuronas. Inmediatamente, teniendo acceso a formulaciones de cannabidiol, 99% puras, de origen sintético, pudimos observar que en estas condiciones donde observamos la sobrecarga de calcio causada por angiotensina en nuestros experimentos celulares, el cannabidiol era capaz de disminuir la concentración de calcio en la mitocondria, la velocidad de transporte de este transportador y, muy importante, la expresión incluso del transportador. Entonces eso fue un hallazgo bastante interesante. Sabiendo que puede tener implicaciones, una molécula que es muy interesante para la comunidad médica y que está utilizándose actualmente en múltiples padecimientos, pues seguimos estudios justamente probando en condiciones de hipertrofia nuevamente nuestras células, nuestros mioblastos, en condiciones de angiotensina, en donde vemos un incremento de la superficie al doble, y fuimos tratando con concentraciones crecientes de cannabidiol, donde vimos y observamos una disminución de la hipertrofia, de los marcadores de remodelación y estrés miocárdico. Teniendo estos datos in vitro, fuimos al modelo in vivo de hipertrofia, en donde fuimos tratando a estos animales con concentraciones crecientes de cannabidiol cada tercer día de forma subcutánea, y pudimos observar en ratones que este fenómeno de remodelación también estaba observándose cuando podemos reducir todo este estrés mitocondrial a través del uso de cannabidiol. Finalmente, no solamente la estructura, sino la función cardíaca, midiendo curvas de presión-volumen, que son gold standard para medir la función ventricular, pudimos observar que el cannabidiol en estos animales era capaz de mejorar significativamente su efecto funcional. Con lo tanto, teníamos datos interesantes in vitro e in vivo, sugiriendo que esta molécula tiene un potencial de traslación. En este momento no nos quedaba más que dar el siguiente paso, que era poder tratar de hacer un estudio clínico. Una universidad pues es incapaz de poder hacerlo, de tal forma que tuvimos que asociarnos con empresas. Particularmente empezó nuestro periodo ya más de emprendimiento, donde empezamos en una colaboración con industria, una compañía canadiense que se llama Cardiol Therapeutics, que tiene las patentes para la síntesis química de cannabidiol. No es el cannabidiol extraído de plantas, sino sintetizado en reactores con formulación farmacéutica. Firmamos un convenio de colaboración. A partir de ello creamos una startup que se llama Nano for Heart, que nos permitió justamente apoyar a esta empresa a entrar al mercado. Actualmente la empresa ya cotiza en bolsa de valores en Estados Unidos, y con los datos experimentales generamos un protocolo clínico usando justamente la formulación de esta empresa, en donde tenemos el cannabidiol, que no tiene presencia justamente de otros compuestos como el THC, en donde ya está formulado, ya está acreditado, y a partir de ello pasamos por agencias regulatorias. Esto fue en el año 2020, cuando justamente aparece COVID. Entonces, nuestro plan de estudiar en insuficiencia cardíaca el cannabidiol pues se vio un poco limitado, pero no completamente, porque decidimos hacer un estudio clínico que se desarrolló en México, Estados Unidos y Brasil, que hablaba justamente de evaluar la eficacia y la seguridad de la formulación de CBD en pacientes utilizando COVID, porque era una forma más rápida de llegar a estudios clínicos para estudiar pacientes con riesgo cardiovascular, particularmente insuficiencia cardíaca. Este grupo, este estudio, que terminó reclutando más de 142 pacientes en los 3 países, fue aprobado por las agencias regulatorias de México, la FDA en Estados Unidos y la agencia regulatoria de Brasil. Sin embargo, pues COVID ya no era un tema elemental, y actualmente la formulación está siendo probada en pacientes con miocarditis, y tendremos liberados, esperemos para el próximo diciembre, los primeros resultados de este estudio. Entonces, con ello lo que quise pasar es otra vez un poquito haciendo el ejemplo de cómo investigación básica sobre mecanismos eventualmente puede trasladarse, y cómo la deformación o la formación o el entrenamiento de un científico puede cambiar hacia el emprendimiento en algún punto, permitiendo justamente hacer investigación que va desde el bench puede tener aplicaciones clínicas, como la investigación preclínica es fundamental, y cómo los binomios y la interacción con industria es la única posibilidad realmente de poder hacer estudios farmacéuticos que pueden costar pues millones de dólares. Finalmente, con esto quisiera terminar mencionando que en nuestro trabajo hemos tenido una larga lista de colaboradores internacionales, particularmente en la Universidad de Chile, en nuestro último manuscrito de hipertrofia, Manuel Estrada, pero también en la Universidad Argentina, estamos muy cerca de Houston, con el Hospital Metodista trabajamos mucho, y pues aquí presento los primeros autores clientes de mi laboratorio que generaron los manuscritos que les he mostrado. Y pues bueno, las agencias regulatorias, nosotros tenemos mucho financiamiento, por supuesto, de empresas farmacéuticas para estos desarrollos, y pues bueno, con ello quisiera terminar la presentación agradeciéndoles su atención.
Gracias. Vamos a hacer una excepción, vamos a hacer algunas preguntas en español. Tenemos tiempo, ¿cierto? Porque estamos bien para algunas preguntas, así que
Perfecto.
profesor Sobrevive.
Gracias, Gerardo. En la acumulación de calcio y luego la subsiguiente eliminación de calcio del espacio intramitocondrial está el sodio involucrado como un elemento necesario según el esquema.
Sí.
Y sodio también participa en la regulación eventualmente del pH intramitocondrial, intercambiando por protones.
Sí.
Entonces mi pregunta es si esa movilización de calcio eventualmente depende del pH intracelular. Esa es una. Y la otra es si esta acumulación de calcio intramitocondrial depende también del influjo de calcio desde el espacio extracelular, de alguna manera.
Sí, gracias, Luis. Pues sí, por supuesto que el movimiento de sodio va a afectar, es un intercambiador que mueve 3 sodios por un calcio, o sea, tiene un efecto. Pero lo importante de la mitocondria es que tiene pues, el gradiente electroquímico, que es lo crucial para mantener un gradiente de menos 180 milivoltios. En realidad, el sodio contribuye, pero de una forma pequeña, porque en realidad el bombeo de protones, justamente a partir de la cadena transportadora, es quien se encarga de regular esos gradientes grandes. Entonces, en la mitocondria realmente el transporte de sodio tiene un papel pequeño en la regulación del pH, porque depende particularmente del movimiento de protones que está trabajando al 100. Respecto a la regulación del impacto que tenga el calcio citosólico, por supuesto, en muchos órganos, incluido el corazón, la interacción entre el retículo y la mitocondria es muy cercana, y los canales liberadores en el retículo o el receptor de reanodina es crucial para acoplar esta dinámica. De hecho, se piensa y se ha demostrado experimentalmente que los espacios son prácticamente unos microdominios de calcio en donde es crucial la interacción, sobre todo en las poblaciones de mitocondrias que están más cercanas a la membrana plasmática. No siendo tan importante a lo mejor esa interacción en las que están en las miofibrillas, pero sí las que están en la membrana plasmática parece tener un papel crucial.
Muchas gracias por la presentación.
Gracias.
Yo tengo un poco de interés más en el efecto sistémico, ya que este uniportador tiene un efecto beneficioso cuando es inhibido a nivel de la mitocondria, pero solo en casos patológicos. Entonces me preguntaba si, por ejemplo, una persona obesa tiene sobreexpresión de este uniportador y si al regular esa expresión, aunque sea un poco, puede afectar también el mejor funcionamiento de la mitocondria y una reducción del peso, que sabemos que unas mitocondrias se ponen en cadenillas, entonces pueden favorecer el gasto calórico, etc. Ya, muchas gracias.
Sí, muchas gracias, Paula. La pregunta es compleja. Nosotros hemos estudiado en obesidad este transportador en modelos experimentales e incluso en muestras de seres humanos. La realidad es que no hemos visto en corazón cambios, pero digamos, no hemos explorado y no ha habido tantos reportes en músculo cardíaco. En músculo esquelético hay resultados que sugieren cambios en la expresión, pero lo que sí te puedo decir en temas de obesidad, un tema muy relevante son las modificaciones posttraduccionales. Y hemos identificado una modificación posttraduccional que es la acetilación, que está muy claramente asociada pues, al incremento de nutrientes en obesidad, que parecería regular de forma posttraduccional ese transportador. La realidad es que pues, ese transportador existe en prácticamente todos los tejidos, expresan ese transportador, que sea más relevante o menos, a lo mejor participa en algunas señales como señalización en redox o en hipertrofia, como lo hemos demostrado en corazón, pero no sabemos en muchos tejidos porque es una proteína relativamente nueva. 2010, 2011 fue identificada molecularmente, aunque sabemos su historia por más de 50 años cinéticamente, en realidad todavía sabemos poco. Y sí creo que puede haber cambios en algunos tejidos. En corazón no hemos encontrado nada, ni en tejido humano, ni en modelos experimentales de obesidad.
¿Más preguntas?
Gracias por la presentación. Sí, no soy experto ni en cosas cardiovasculares ni mitocondria, pero me perdí un poco la parte de la conexión entre el uniporte que deja entrar calcio, ¿cierto? Y eso se necesita para que funcione bien. Pero después, en algún momento, eso se traduce después en la producción de ROS, que activa después a NF-κB, fundamental para la respuesta proinflamatoria, ¿cierto? Entonces, como que me perdía un poco por qué ahora este aumento de calcio lleva a ROS, porque si se necesita para que funcione bien la mitocondria, ¿por qué realmente tenemos ROS?
Sí. El fenómeno particularmente es un incremento en la concentración de calcio por arriba de lo fisiológico, permite generar un factor o una condición que se llama transición de la permeabilidad de la mitocondria. Esto implica que transportadores que estaban en la mitocondria teniendo su función normal, al unir grandes concentraciones de calcio, lo que hace es que esos transportadores se vuelven en canales inespecíficos que ahora ya no permiten la permeabilidad selectiva. Al no tener permeabilidad selectiva, la cadena transportadora empieza a trabajar con mucha mayor velocidad para tratar de compensar la pérdida de ese gradiente, y es ahí donde muchos más electrones se empiezan a fugar y empiezan a acumular y formar el radical superóxido. Entonces, este fenómeno en condiciones fisiológicas se sabe que ocurre esta apertura transitoria de estos poros, como un tipo de flickering, pero después, cuando se perpetúa grandes concentraciones de calcio, empieza a aparecer el estrés oxidante. Estos poros se permanecen abiertos y se hacen megacanales que permiten pues, salida y movimiento de sustancias, la liberación de citocromo C, pero el estrés oxidante tiene que ver con que trata de compensar la mitocondria la pérdida del gradiente, moviendo más electrones y por lo tanto incrementando el radical superóxido. Ese es el
Hola, gracias por la charla. Te quería hacer una consulta. ¿qué está sucediendo en este momento con NSLX? Porque últimamente se ha descrito y ha estado bastante en boga MCU en el contexto del músculo esquelético, el músculo cardíaco y también en la parte neuronal. Sin embargo, de NSLX se sabe poco, muy poco, casi nada diría yo. Y a las finales el mayor problema es la acumulación de calcio mitocondrial, que genera un problema extraoxidativo, bla, bla, bla. Pero hasta el momento lo que sabemos es que MCU es el principal factor que podría estar contribuyendo a esta sobrecarga mitocondrial. Pero resulta que NSLX es un intercambiador que podría contribuir también y podría ser un blanco terapéutico maravilloso. Entonces, no sé qué hay recientemente, no sé si ha habido más investigación al respecto, no sé si suena una propuesta interesante a seguir investigando. Eso quería saber.
Sí, muchas gracias. Yo creo que igual contigo coincido de que hemos visto mucho el sistema de entrada, pero el sistema de salida no ha sido tan enfocado también porque ha sido difícil identificar los factores moleculares que constituyen este transportador. Mi respuesta sería en dos sentidos. Igual que pasó con el MSU al principio, la deleción de estos transportadores no tenían fenotipo, probablemente porque están relevantes estas proteínas que de alguna forma se compensan con otras moléculas. Pero cuando se han hecho deleciones ya en animales adultos, específicas cardíacas, por ejemplo, la hipertrofia y la mortalidad en esos animales es enorme. O sea, el quitar ese transportador parece que también está contribuyendo importantemente a las dinámicas de calcio y la mitocondria, un poco opuesto a lo que nosotros vemos, pero es clarísimo que hay una hipertrofia y una mortalidad en los ratones cuando adultos tú silencias el intercambiador. Nosotros tratamos de hacer estos experimentos ahora sobreexpresando un poco para ver efectos y no observamos un fenotipo, pero lo hemos hecho solamente en mioblastos cardíacos, no lo hemos hecho en células, digamos, un poquito más maduras. Pero sí la literatura consiste en que probablemente no sabemos completamente todos los elementos que juegan en la dinámica de calcio mitocondrial, pero sí completamente de acuerdo que tenemos que estudiar mucho más el intercambiador.
Sí, gracias. Espero no me mates después de esta pregunta. ¿Qué se tiene pensado como para extraer el MCU cristalizado con rutenio 360? En tu laboratorio hay una estrategia como para caracterizarlo ya con este antagonista completo del MCU.
La realidad es que yo no hago nada de cristalografía ni estructura de proteínas. Lo que se hace, ¿cómo? Purificarlo. No, lo purificamos con anticuerpos. Nosotros hacemos mucha inmunoprecipitación con el anticuerpo. Así es como estamos siguiendo las modificaciones posttraduccionales. Nosotros unimos anticuerpos específicos y inmunoprecipitamos. Lo que estamos haciendo muy finamente, o queremos hacer muy finamente, es la caracterización en forma de canal iónico en mitoplastos para poder ver características. Además hemos hecho modificaciones del rutenio con algunos otros componentes que lo pueden hacer más o menos selectivo, más o menos permeable. Entonces, por ahí las caracterizaciones electrofisiológicas en el propio canal, eso sí estamos queriendo hacer, pero no sobre cristalizar y generar temas de estructura en el transportador, no es nuestra área de interés ni expertise.
Bien, vamos a dejar hasta acá las preguntas. Agradecer de nuevo al doctor Gerardo Rivas y quisiera hacer solamente un comentario que sea felicitarlo por la cantidad de trabajo, de avance que ha mostrado desde su visita anterior a nuestro programa. Es impresionante lo que han avanzado a la medicina translacional, así que los felicitamos y esperamos que podamos colaborar en nuestro programa.
Claro que sí, será un gusto.
Así que felicitaciones de nuevo, un aplauso.
Muchas gracias.
De acuerdo al programa, tenemos entonces 30 minutos de café, que sería en el hall de la Escuela de Tecnología Médica. Así que nos juntamos acá, de vuelta a las 11, el café es acá al lado, en el hall de la Escuela de Tecnología Médica.
Gracias.
2022 assistant professor at the Integrative Physiopathology Center at the University of Valparaiso. Between 2017 and 2022, he was part of several grants in which he studied the interaction between skeletal muscles and microbiota. And today, he will present his work related to the role of gut microbiota in obesity. I have to say that since 2023, Dr. Jorquera has been held a position of assistant professor at the Institute of Nutrition and Food Technology at the University of Chile. The same year, in 2023, he achieved a milestone grant awarding by leading the only Latin American project awarded by the dynamic, in which he will study the Dynamic Resilience Initiative. The grant was called the Dynamic Resilience Initiative of the Welcome Leap Program. He was awarded this grant by studying the relationship between the microbiota, the gut microbiota, and skeletal muscle function in the elderly. So today he will talk about the role of the gut microbiota in obesity and the potential microbiota-based therapeutics for obesity management. Please.
Thank you very much for the invitation. It's a pleasure to be here. And this morning I want to tell you about the relationships between gut microbiota and the regulation of the metabolic function. I want to tell you that like in a history point of view, the main articles that have been describing the relationship between changes and modulations in gut microbiota and the metabolic function and the pathophysiology of obesity, and also discuss a little bit about interventions that have been done in humans for the management of obesity. So in the beginning, I want to define what is the gut microbiota. Gut microbiota is the collection of bacteria, archaea, eukarya, and also virus that are colonizing our gastrointestinal tract. These microorganisms have co-evolved with us during thousands of years and have established a symbiotic relationship with us. So now we know that changes in gut microbiota can have profound effects in several contexts, in several diseases, and this is the thing that we are going to be discussing today. For example, this is a very early work in the field in which researchers study the role of gut microbiota in the regulation of metabolic function. And they work with germ-free animals. These are mice that were raised in a complete isolation facilities, so these mice have no bacteria in their bodies. They are completely free of gut microbiota. And they also have conventionally raised animals, which are control animals. And they also have animals that were raised, for example, like germ-free animals until adulthood, and then they receive fecal microbiota transplantation from a donor being adult. So they receive late in life the bacteria in their intestine. And in this paper, authors evaluated the body fat percent, the epididymal fat weight, and as you can see, animals without a gut microbiota have lower body fat and epididymal fat in their bodies compared to animals that do have gut microbiota. They also evaluated leptin levels, insulin levels, and glucose levels in blood, and as you can see, animals without bacteria have lower levels of these elements in blood, and they evaluated the metabolic function through glucose tolerance and insulin tolerance tests. And you can see the glucose levels in both tests were always lower in animals without bacteria. So this very early work show us the importance of the presence of or absence of gut microbiota in the regulation of metabolic function. In the next article, I want to show you that researchers continue working with animals without gut microbiota, here germ-free animals. They continue working with animals that receive bacteria during adulthood, but in this case, animals were fed with a Western diet, rich in fat and sugar. And you can see here that animals that have gut microbiota here experience an increase, a significant increase in body weight compared with animals that do not have any bacteria in their bodies. So again, we are seeing how the presence or absence of gut microbiota is very important in the context of the metabolic function. And also, authors evaluated, for example, intracellular metabolic signal. They evaluated the phosphorylation of AMPK, which is a kinase very important for several functions in highly metabolic organs. They evaluated the activation of AMPK in skeletal muscle and in liver. And you observe here that in the presence of gut microbiota, the phosphorylation of AMPK is quite reduced compared to that phosphorylation observed in animals without a gut microbiota in the context of a Western diet. So this is very interesting because it shows like changes in gut microbiota could have a specific effects in highly metabolic organs that are regulating our complete metabolic function. Today, we know that there are axes. For example, I show you here the gut-liver axis, the gut-muscle axis. These are bidirectional communications between the gut and the gut microbiota with highly metabolic organs. And it has been shown that alterations in this axis are associated to the development of some diseases. For example, in the case of gut-liver axis, alterations in this signaling is associated to the development of non-alcoholic fatty liver disease, to the development of fibrosis, cirrhosis, and in the case of the gut-muscle axis, alterations in the communication between the gut microbiota and skeletal muscle is associated to alterations in insulin resistance, in activation of atrophy and sarcopenia in the skeletal muscle. This article is very interesting because researchers here evaluated the composition of gut microbiota in two populations, in lean individuals and obese individuals. This study of the composition is done by the sequencing of the genome, the bacterial genome, And most commonly, this sequenciation is done in the 16S rRNA gene, which is a gene that have highly variable zones. So through this sequenciation, you can know what type of bacteria you have in your sample. So here we have a two-component plot, and each point is one sample of the gut microbiota, and the closer the points, the more similar are the bacteria in the gut in the sample. So you see here that in lean mice, the microbiota is quite similar, but it's very different to the gut microbiota observed in obese animals. So we know that gut microbiota is changing when an individual is experiencing obesity. They also evaluated the two main phyla of bacteria, the two main types of bacteria in the intestine, and they observed that the level of Firmicutes is overrepresented, and the level of Bacteroidetes is lower in obese mice compared to lean animals. So they have found specific changes in the bacteria living in the colon. And this is a very nice experiment done in humans. And you can see here they have lean individuals, they have here obese individuals, and the changes in the proportion of Firmicutes and Bacteroidetes is similar in humans to what was observed in mice. Because as you see in obese individuals, the levels of Bacteroidetes is reduced compared to lean individuals, and the level of Firmicutes is overrepresented. And this study is very nice because these obese people were under a low-calorie diet over a course of 1 year. So as you can see, as the time passes, the representation of Bacteroidetes increases and the representation of Firmicutes decreases. And at the end of the treatment of this low-calorie diet, the profile of the gut microbiota in obese individuals is similar to that observed in lean individuals. This is very interesting because it shows like dietary interventions could modulate gut microbiota, and maybe the positive effects observed by dietary interventions in obese patients could be associated to changes in bacteria living in our intestine. This is another very nice article because it shows how is the similarity of gut microbiota between individuals. Here in this plot, it shows the lower numbers here. It indicate more similarity of the gut microbiota between two samples. So here you see when researchers compare your gut microbiota with yourself, so the similarity is very high. And here is when you compare your gut microbiota with an unrelated subject, and the gut microbiota is quite different. But they also study the gut microbiota between twins, monozygotic twins, and this is nice because these people have the same genetic background. So you can see that the gut microbiota is more similar between twins if you compare someone to an unrelated individual, but it still is quite different to when you compare the gut microbiota to yourself. So the genetic background is important for establishing a defined pattern of bacteria living in your colon, but definitely there are other factors that are involved in the acquisition and specific gut microbiota in each individual. And we know today that the main factor that is regulating what kind of bacteria we have in our gut is the diet. I mean, the type of diet we are eating. Another important result that the researchers obtained here is that they evaluated the diversity of bacteria living in the gut. You can see here that the diversity of the bacteria in obese individuals is reduced compared to lean individuals. This is interesting because we know today that in many diseases there is a reduction in diversity of bacteria, and actually the diversity of bacteria living in our gut is associated to a healthier state in the host. So a goal for improving the health state in the host could be promoting the diversity of bacteria and the representation of good bacteria living in our colon. Here we have a summary of many factors that can promote a healthier profile of the gut microbiota, and many factors that are associated to a risky profile for developing alterations like insulin resistance and obesity. Here, for example, we see that the type of fat we eat is very important. For example, omega-3 polyunsaturated fatty acids are associated to a healthier profile in the gut microbiota. The consumption of complex carbohydrates rich in fiber, the consumption of vegetables, fruit, some type of diet, some drugs like metformin or GLP-1 analogs could be associated to positive effects in the gut microbiota. But we have other factors that are associated with a risky profile of bacteria. For example, the consumption of saturated fat from animals, the consumption of refined sugar, the consumption of, for example, food rich in fat, in sugar, the low or few consumption of vegetables and fruit. I'm going to show you some results so we can understand these regulatory functions of this kind of food over the gut microbiota. For example, this article showed the effects of high-fat diet in mice. But we also evaluated the effect of the high-fat diet with different type of fiber. They have non-fermentable dietary fiber and they use fermentable dietary fibers. Okay? And they evaluated several kind of bacteria in the fecal samples of the mice. They evaluated Bifidobacterium, they evaluated Eubacterium brutali, Lactobacilli, Bacteroides, many other bacteria. And they observed a very nice pattern here in the Bifidobacteria, in which they observed that when animals were eating a high-fat diet, the percentage of Bifidobacteria is reduced in the gut microbiota. But if you give to these mice the high-fat diet, but for example the non-fermentable dietary fibers, the representation of Bifidobacteria increases, and even in the presence of fermentable dietary fibers, the representation of Bifidobacteria increases even more. And they observed that they evaluated the levels of endotoxin in blood, and they observed that endotoxin is increased, as you can see here, in mice that have lower levels of Bifidobacterium in the gut microbiota. Remember, endotoxin is LPS, is a lipopolysaccharide, which is a very immunogenic molecule that is present in the cell wall of some bacteria, and LPS can cross to the bloodstream and can activate an immune response, and this immune response leads to inflammation, and inflammation can alter metabolic function. And actually they observed that higher levels of endotoxin in mice is associated to a poor management of glucose in a glucose tolerance test, Higher levels of endotoxin in mice is associated to elevated levels of fasted insulin, elevated levels of body weight, and elevated levels of visceral adipose tissue. We know now that alterations in gut microbiota, which is called dysbiosis. Dysbiosis is a term that it simply means an alteration of the bacteria living in the intestine compared to a control condition. And dysbiosis is associated to a condition called like leaky gut, which is a leaky gut. Is a gut that have increased its permeability, so elements that usually are unable to cross to the bloodstream can cross and can activate some responses, for example LPS, flagellin, or other molecules from bacteria. And this can cause inflammation, and this inflammation is related to the development of metabolic diseases. This is another example of how diet can regulate gut microbiota. For example, in this paper, researchers evaluated two different kind of fat. They evaluated animal fat, like lard, which is a type of fat rich in saturated lipids. And they also evaluated fish oil, which is a type of fat rich in polyunsaturated lipids, a healthy lipid. And they observed, for example, that animals that receive a diet rich in lard increase their weight gain in a significant way compared to animals fed with a fish oil. And they evaluated the gut microbiota, and here we have again a two-component plot, and you can see that the microbiota is quite different between these two groups of animals. And here we have the main types of bacteria overrepresented in animals that were fed with lard, and here we have bacteria that are overrepresented in animals that receive fish oil. Remember, please, this bacteria, Akkermansia municiphala, because this bacteria is very interesting. This bacteria is overexpressed in animals that receive fish oil, because this bacteria, Akkermansia, it has been associated to beneficial effects in metabolic function in animal models and humans. Also, they evaluated the activation of Toll-like receptor signaling. They observed that lard increases the activity of the signaling that is associated to inflammation. And if they use germ-free animals, I mean animals with no bacteria, they observed that the effects of this diet rich in saturated lipids have lower negative effects. They measure weight gain. Here it was reduced the negative effects of lard if you don't have any bacteria, and the activation of inflammatory cells, it was also reduced. So this is shows us how dietary interventions are regulating gut microbiota, and this can explain, at least in part, alterations observed in metabolic function. Here we have the role of Akkermansia municiphala. As I was telling you, this bacteria has been associated to many positive effects in animal models and humans, and it has been proposed that can reduce the development of some important diseases associated to metabolic alterations. And here I show you a very nice work in humans. This is a work done in overweight and obese individuals, and they receive daily over 3 months a supplementation with Akkermans as a probiotic. So they have 3 different groups. They have the placebo groups, they have a pasteurized group, which means that the bacteria is killed by hot, and they have the group with the live bacteria. And you observe here that the insulinemia in patients over the course of 3 months in overweight and obese individuals was reduced, the fasting insulinemia. They also observe a reduction in insulin resistance in people who received the pasteurized version of Akkermansia and the live bacteria. And they observe also that the insulin sensitivity is increased in people who receive the pasteurized version of the bacteria. They also evaluated the total cholesterol levels and the LPS in blood, and as you see here, they observe a reduction in cholesterol levels in people that receive Akkermansia in a pasteurized version, and LPS also in blood was reduced. They also evaluated body weight, they evaluated fat mass and hip circumference, but they didn't see any change, but they see important trends here in people receiving the pasteurized version of the bacteria. It is important to know that the main results that they observed here, it was in people that received the dead bacteria. So that means that some components in the bacteria could promote a modulation of gut microbiota and have these positive results. But an open question is how bacteria, how if you modulate the bacteria in your gut can promote alterations in the metabolic function. So there are many theories and many potential metabolites that can be secreted by bacteria and have positive effects in the host. One of these metabolites is short-chain fatty acids. Short-chain fatty acids are molecules that are produced for the fermentation of fiber in the colon, and some bacteria can produce them. Also, bacteria can modify bile acids, so it is proposed that these bile acids can have positive metabolic effects. And many other options had been proposed for this crosstalk between the gut and metabolic organs. I'm going to talk about the short chain fatty acids. For example, you see here how these molecules like propionate, acetate, and butyrate have specific G-coupled receptors, and they also even can enter to the cells because they have transporters. And these molecules can have positive effects in the adipose tissue, in the skeletal muscle, and in the liver. So the main metabolic organs that are regulating several functions and that have alterations during metabolic diseases. For example, in the adipose tissue, short-chain fatty acids can increase lipid storage capacity, can increase the adipogenesis, can reduce the production of pro-inflammatory cytokines and chemokines. In skeletal muscle, short-chain fatty acids can promote glucose uptake, can promote insulin actions, can induce fatty acid oxidation, among other functions. And in the liver, short-chain fatty acids can induce gluconeogenesis, can reduce lipid storage, can induce fatty acid oxidation as well. So it is proposed that these short-chain fatty acids have, in general, beneficial effects in highly metabolic organs. So in this paper, these authors propose to give propionate to overweight individuals. So they give a supplementation of this postbiotic. Postbiotic is a molecule that is produced by bacteria, and you can use them for biomedical use. So they give this supplementation for 24 weeks, and they have a group that receive 10 grams per day of the propionate associated to a type of fiber, and they have the control group that receive the fiber without the short-chain fatty acid. And as you can see here, they observe that people that receive the propionate have a reduction in energy intake. They also have increased production of PYY and GLP-1, which are incretins that are associated to better insulin response. And they observed that people that gain weight during the study was mainly in the group of people that it was not receiving the propionate. And they also observed that people who receive propionate experience a reduction in intrahepatocellular lipid content. So it is a promising results in the use of this postbiotic. And finally, I want to show you the type of intervention that is called the fecal microbiota transplantation. This intervention means that you have a lean donor, in this case, in the case of this, you have a lean donor, and you have the feces of this lean donor, and you have a process when you obtain the gut microbiota from the donor. Then you can lyophilize this bacteria, and you can encapsulate the bacteria in capsules like that. So the people who receive the fecal transplantation take some of these capsules, and this research was done in patients with severe obesity. And as a prebiotic, researchers complement this fecal microbiota transplantation with high fermentable fiber in one group, or with low fermentable fiber. And we can see here that people who receive the FMT, the fecal microbiota transplantation, with low fermentable fiber have a reduction in insulin resistance, increase in insulin sensitivity. They have reduction in insulin levels, in fasted insulin levels. They also have an increase in GLP-1 production or presence in blood, a reduction in insulin in blood in a glucose tolerance test, and they have a specific representation of bacteria that are associated to positive metabolic effects. So we have many results that are showing us how important is gut microbiota in the regulation of metabolic function. I show you here my project. I have a project in which we are going to modulate gut microbiota in animal models, but also in people. We're going to perform fecal microbiota transplantation from young and trained donors, because we want to improve the diversity of gut microbiota in these all individuals. We're going to work with all animals and with older adults. And we want to change and improve the gut microbiota in these individuals to improve the resilience capacity. So we are going to study several different types of stress. One kind of stress that we are studying is the metabolic stress and obesity. And our theory is that the modulation of gut microbiota is going to improve the resilience capacity in older adults, and they are going to improve or at least retain their metabolic function, their cognitive function, and their muscle function when they are exposed to an stress event. So these are my collaborators, and this is my team. So the conclusions, there there is not one species that can explain the development of obesity or the prevention of gaining weight. But with the evidence I show you, we can say that it's evident that gut microbiota plays a very important role in the development of metabolic disorders and obesity, and the modulation and intervention in gut microbiota could be very potential therapeutical strategies to improve the metabolic functions in overweight and obese individuals. And for the modulation of this bacteria, we can use dietary interventions, prebiotic, probiotic, fecal microbiota transplantation, postbiotics. So we have many options. And we need more research because, as I show you, the gut microbiota is quite different between us. So the precise type of therapy probably is very personal for each patient. Thank you very much.
We have some minutes for questions.
Thank you very much. Can it be in English or Spanish?
Como tú quieras.
Okay, in Spanish. No, it's all right. I have 2 or 3 points, very short and simple points, I would say. One is the significance of the gut microbiota in obesity, as you're mentioning here. But many patients with obesity are also showing type 2 diabetes mellitus. Is that something that you have considered in your future projects or actual projects? And that's one. And the other thing is whether you know about mental health and gut microbiota.
Yeah. Well, in the first question, yes, I didn't show you here, but there is strong evidence that gut microbiota has a function in the development of type 2 diabetes, gestational diabetes as well. So definitely a potential therapeutical option could be the modulation of gut microbiota in those contexts as well. And the second question, yes, there is a very important axis, maybe the most studied axis of all this, which is the gut-brain axis. So it has been reported that people with psychiatric diseases or mental health alterations have dysbiosis. And it is there are published very interesting articles showing in animals and humans how modulation of gut microbiota can improve mental health. Actually, it was published very recently, like last year, I guess, psychobiotic diet, which is a diet based in fermentable food. So this type of diet show beneficial effects in patients with depression.
Gracias. ¿Se sabe si esta microbiota y el rol que juega en los desórdenes metabólicos, hay diferencia entre machos y hembras? Y si es que tú vas a considerar eso en tu estudio.
Sí, hay diferencias. De hecho, nosotros empezamos a trabajar en mi proyecto de iniciación con donantes machos, eran ratones machos que donaban, con el cual hacíamos el trasplante de microbiota fecal a ratones envejecidos. Y los receptores eran ratones machos y hembras, y en los ratones machos veíamos efectos bastante claros y en hembras veíamos una dispersión enorme. Y hay publicado, hay un concepto que se llama el gender microbiome, que es en la representación bacteriana entre hombres y mujeres tiende a ser distinta. Y es por eso que en el proyecto nuevo que tenemos ahora y en el que vamos a realizar con personas, vamos a tener donantes hombres y mujeres para los donantes hombres para receptores hombres y donantes mujeres para receptores mujeres.
Thank you very much for your presentation. As you said before, there is a correlation between the gut microbiota disorder with psychiatric disorder. Under that point of view, there is any correlation or study that said what happened with people with eating disorders, for example, microbiota dysbiosis, or there is any diet recommended for that population?
Yes, actually, for example, in Parkinson's disease, it is proposed that the first alterations that people with Parkinson have is not cognitive or the tremors. The first alterations is gastrointestinal. So they can have diarrhea or they can have constipation. So it is proposed that there is a theory that the disease is beginning there in the intestine and can be affecting other organs with the passing of time. So there are several publications trying to identify biomarkers in the gut microbiota to identify very early stages of some neurodegenerative diseases.
Thank you for the presentation. I have a question related to in the first part of the presentation you showed that Firmicutes and Bacteroidetes, they are present in this gut microbiota. And I wanted to ask you if maybe are you also planning to go further maybe in the study with this kind of dark matter of the microbiota, which is high diversity but low richness? Maybe these bacteria are having an effect. And the other question is, which could be the functional role or functional role in the gut?
Well, this classification is very broad. It's a very general classification because Firmicutes and Bacteroidetes are the main two phyla of the bacteria. So inside this group you have many bacteria. So these results I show you is, there is a difference between these two groups in lean and obese individuals, but now the research is oriented to identify specific differences more to the general level, to the species level, and avoid these high groups because it is difficult to intervene in patients.
Yeah, very interesting talk. So you showed at the beginning a slide where essentially germ-free mice didn't become obese, right?
Yeah.
Compared with the ones that had. So, this is a bit of a provocative statement now. So why don't we just treat obese people with antibiotics, get rid of all the bugs, and then give them a good microbiota? Theoretically should solve the problem, no?
Yeah. Actually, the antibiotics is the use of antibiotics is associated to a risky profile in the bacteria. So probably you are more in risk to develop obesity if you use antibiotics, and you can have very negative consequences because you can have resistance. You can develop resistance to this antibiotic, and even you can create niche for very infectious bacteria that can reach to the intestine, like Clostridium difficile. So it's not a good option to use antibiotics. It's better to modulate the gut microbiota to enrich the bacteria that are good for you and reduce those that are not good for you.
And what about the short-chain fatty acids? I mean, what exactly do they do anything specific to the microbiota?
Yeah. Well, I show you the effects of short-chain fatty acids in highly metabolic organs, but short-chain fatty acids can for example, also regulate the immunitary function. So these molecules can improve the function of immune cells and can reduce the presence of negative bacteria and improve the presence of positive bacteria to the host.
The last one.
Thank you for your presentation. Thinking in a translational point of view, does fecal transplantation maybe thinking as a therapy? What do you think?
Yeah. I mean, it's a potential therapy. There are many clinical trials doing it. I mean, they have shown very good results. You have to be careful with the donor. The donor has to be checked with several diseases. It has to be a very healthy person. But yes, it's a very good option. Well, we have to be careful with that because you have in the recipients different type of bacteria representation. So probably you're going to have more effects in some group of people and less effects in others. So probably the intervention in the bacteria in the colon is going to be more personalized with the time.
Well, thank you very much. We give an applause again, Dr. So we have to continue with the program. Let me see. So our next speaker is Rodrigo Chamorro. Rodrigo received his master's degree in nutritional sciences from the University of Chile. And then he received his PhD degree from the same university under the supervision of Dr. Patricio Beirano. Afterwards, Dr. Chamorro was awarded a fellowship of the Latin American Nutrition Research Leadership Program of the Latin American Nutrition Society. Since 2016, he has been assistant professor at the Department of Nutrition, Faculty of Medicine, University of Chile. In the same year, he received a fellowship for a postdoctoral position at the Center of Brain Behavior and Metabolism at the University of Lübeck in Germany. His main area of expertise is chrononutrition associated with human obesity, and today he will present his talk entitled the impact of chronodisruption on obesity and obesity-related metabolic alteration. Thank you very much.
Thank you very much for your kind invitation. Rodrigo and the group, you are doing a very nice job here with this event, so thank you again. This is my first time here in the University of Talca and also interacting with people from the ACTIS, so I'm very happy to be here. I will be starting to work this year with Mariana in line number 7 of this research effort. So I'm very happy, and today I will show you part of my previous research focus over the last 5 years or so. Then my idea was to introduce you to this topic that with Mariana we hope to be starting to work with over the next 2, maybe more years, I hope so. Briefly as an outline, I will give you just a brief introduction because I considered it was necessary to go through to circadian rhythmicity, then focusing on the role of or the potential role of chron disruption, of disrupting these rhythms, and to see whether we have metabolic or organ consequences, and obesity, of course, with the focus on, and then the potential restoring effect of timing interventions, particularly with diet, is my main focus, but also with other factors, and finally some perspectives. So first, very briefly, we live in a 24-hour context, so that is our time frame to behave, and that's very, it can be easily overlooked through the days. So as you can see, many processes and states like sleep, wakefulness, endocrine rhythms, body blood pressure, body temperature reaches its highest point or its highest activity in specific time points of the day. This is mainly, as you can see here, this is daily changes, so it can be a proper circadian rhythm which has a duration of 24 hours, but other characteristics, but can also be consequences of our behavior. So we are continuously eating, moving, sleeping, so that behavior can also drive some of these daily changes. So it has to be a separation between those two. So as you can see here, for example, circadian rhythms are very well known in several aspects of our physiology. As you can see, for example, for melatonin, cortisol, and blood lipids such as triglycerides, we have a clear increase during the night, for example, specifically for melatonin and blood lipids, and cortisol peaking right after we awake in the morning, and then with the highest level during the day, but decreasing just before sleep. So it has to be a temporal order in these several aspects of our physiology to have a proper functioning over the day. And on the other hand, motor activity, of course, which is the other one. Motor activity is the highest over the day in our case because we are diurnal mammals. So this leads us to a temporal context which leads to a temporal order in our physiology, and that is the main consequences of having this circadian rhythmicity in us. So whether these rhythms are circadian or just fluctuations in physiology, that it has to be defined under strict controlled conditions. So in that context, it is now known that the molecular mechanism underlying the circadian rhythmicity is currently well described. So I'm showing you here to the left the central clock genes, which are called a set of clock genes, which are organized in as you can see here, in a translational feedback loop over the day, with this self-regulation leading to a period of 24 hours, roughly. And then, so we have these two clock genes, clock and BMAL1, leading the transcription of two sets of periods and cytochrome genes 1 and 2, which in turn inhibits later during the day the transcription of the first part of clock genes, clock BMAL. Additional loops here, shown here below, give us to the system stability or further stability, I will say, with some nuclear factors, which are the ROR and red ERB alpha, having a positive and negative influence back to the clock. So graphically, you can see also here, this is the BMAL clock heterodimerization in the nucleus, lead the transcription of this period and cytochrome genes, leading to the translocation to the cytoplasm, where they get phosphorylated and then inhibiting the central expression of body clocks. So this machinery, I would say, leads to a 24-hour organization in several processes, such as food intake, fasting, sleep, awakening, activity, rest, of course, endocrine rhythms, and so on. So this is the potential relevance for us. So now we know that the circadian system is composed by a clear structure within our body. So we have a central clock which is located in the nucleus, suprachiasmaticus nucleus in the hypothalamus, for which light, the alternation in between light and darkness is the main stimulus for the organization of this central clock machinery. But then in peripheral organs such as the stomach, adipose tissue, liver, gut, as previously Gonzalo was talking, pancreas, the heart, and the adrenal gland, they all have the same molecular structure. So every day they are having these 24-hour expression rhythms. So there's a connection in between those two. So the central level and the peripheral level, they are connected. They are proposed to be connected through the autonomic nervous system and humoral factors or endocrine factors such as cortisol and melatonin rhythms, leading to the connection in between the environmental part through the light with the internal part, which is the connection between the central clock and peripheral ones. And below, here you can see that food intake and exercise are proposed as two very relevant factors in training the clocks. But in training means that it's the stimuli to start or for the offset of the expression at these peripheral organs. So now we know that light is the most important factor for the central clock, but food intake and activity or exercise is even more relevant for the peripheral ones. So that is very interesting in the context of metabolic research right now, and I think that is a very hot topic regarding obesity research. So that leads us to the effect of chron disruption. What is the metabolic consequences of altering these rhythms, and then what can tell us about the potential effect in obesity research? So first, I would like to say that when an organism is well entrained to the environmental factors, such as I already told you, we can expect that the central clock is entrained every day, first, and then you can expect that it's a phase match in between environmental factors and social factors, such as the daily rhythms the animal possess during the day. And that could lead to metabolic homeostasis or normal metabolic regulation. On the other hand, when there's a mismatch in between those factors, arising, for example, as you can see here, right in the upper right, so for example from shift work, erratic meal timing, social jet lag or jet lag itself, and erratic light exposure during the day, that could lead to this mismatch in between social and environmental clocks, leading to metabolic dysfunction. At the molecular level, what we could see is that circadian disruption lead to decreased amplitude of clock genes oscillation, and also a loss of synchrony between central and peripheral clocks, and also to a phase mismatch at in between peripheral organs. So the proposal now is that misalignment clocks can lead us they are very indeed associated with rhythms alteration, of course, and that is associated with several chronic diseases, both metabolic ones, but also mental disorders such as depression, cardiovascular diseases, of course, obesity, type 2 diabetes, cancer, fatty liver disease, and inflammation. So it's a whole set of metabolic consequences, I would say, that we could expect from this. So as I told you, several factors can be in charge of this disruption of rhythmicity, and I will go through some of them. I hope briefly. So first, light. Light at night is supposed not to be happening, but we are under huge light contamination every day. This is a summary in animal models, and here to the left you can see that we can use constant light in animals experiments, or we can use dim light, which is a sort of mild light exposure, or we can see another type of shifting paradigms. So under these conditions in animal models, the evidence, as you can see here in this column highlighted in red, that mainly there is a attenuated rhythmic expression of several clock genes at the central level. And to the right here you can see that regarding endocrine and behavior effects, so mainly regarding corticosterone or cortisol, an abolished rhythm, as I show you first in the early slide. Regarding behavior, with contact with both constant light or even bright light or dim light, the animal turns to be arrhythmic mainly, or with an alterated rhythmic activity over the day. So this is not news. We are contaminated by light. This is satellite data from a couple of years ago, showing that more than 80% of population lives in areas with more light exposure at night than the desirable. We are not the exception, of course, and so that is associated, as I show you here, in epidemiological studies showing that light exposure at night in this huge sample of Chinese participants is associated with metabolic alterations, specifically glucose homeostasis. As you can see, for example, HbA1c levels, fasting plasma glucose, 2 hours plasma glucose after a tolerance test is slightly associated with an increased risk of alteration in this population. This is not only the case for type 2 diabetes, it's also for hypertension, obesity, and cardiovascular disease overall. So in the laboratory, researchers can expose humans to light, very bright light, for example 4,000 lux as compared to a 10 lux. Now we are, for example, in a fluorescent lamp, we have some like 350 lux, and that's the idea. So in this context, they exposed to participants, they were healthy, to this bright or dim light conditions. And as you can see, for glucose response after breakfast, for triglycerides response after breakfast, and for heart rate variability, after breakfast. So regarding glucose and heart rate variability and heart rate itself, it doesn't matter the light exposure in normal weight individuals. But the author found that there's an increase in plasma triglycerides after the same breakfast under the bright light exposure. But here below you see the same protocol, but in individuals with obesity and type 2 diabetes. And as you can see, there's a clear different response in under the bright light exposure regarding blood glucose, with an intolerance shown by the subject. Also the postprandial response of triglycerides and heart rate. So when the metabolic disease is already installed, it seems to be the further the damage the circadian disruption can have in this population. So this is also true for blue enriched light, which is the light that you can obtain from these electronic devices that we are currently using. It doesn't matter when you receive the light. This is the light in the morning or in the evening here, and the authors here evaluate the insulin resistance in participants over 3 days with 370 lux. As you can see, whenever we are exposed to blue enriched light, insulin resistance goes high. We tried to approach this effect, and we did this experiment over 2 days, but we were wondering whether light exposure, even in a minor level, which is dim light, you can see you cannot see, but it was 5 lux, so almost darkness, whether that has an effect on metabolic and sleep regulation. And we saw that no. So very light, a mild dim light exposure it has an effect on glucose response, insulin response, or insulin secretion. In these subjects, they were all normal, healthy ones, young, but the sleep quality was affected. So 2 nights of very dim light reduced sleep quality as assessed by sleep analysis of the EEG. And also you can see the cortisol levels in the afternoon and right in the morning after awakening, which is a clear indicator of alertness, among others. It was reduced after just 2 nights of very mild light when subject was already sleeping. So then we switch on the light. So it seems to be a clear effect of light exposure in humans regarding metabolic consequences, which is depend on the stimuli. Regarding sleep, sleep is one of the states which is commanded by the circadian clock, but also goes back to the circadian clock, so influencing back this rhythmicity. So sleep is a state which is has a circadian regulation, but also a homeostatic regulation. Also, the social interaction will influence our sleep organization. So this the question was, okay, first, this is not news. This is for several decades ago, showing that when we are able to sleep for 4 hours a night, there is a clear metabolic consequence, showing increased insulin resistance, increasing cortisol levels in the afternoon before sleep, reducing growth hormones, of course, decreasing leptin levels, and increasing the sympathetic nervous system tone. So just 5 days sleeping 4 hours a day leads to these acute metabolic consequences in humans. We did an experiment a couple of years ago showing that the effect of sleep restriction, so sleeping 4 hours of sleep deprivation. We want to see if there was a differential effect. So as you can see here, both conditions, but mainly sleep deprivation, decreased insulin, fasting insulin, fasting C-peptide, and reduced insulin sensitivity compared to a control condition of 8 hours of sleep, also increasing some fat molecules involved in fat metabolism, increasing after sleep deprivation. But we were also able to show that this very nice effect that under control condition, when we are able to sleep 8 hours, there's a clear 2 distinct peaks on gene expression in the fat biopsies that we took from the abdomen in the morning and in the evening. So the left peak here is the number of genes that is more expressed in the morning, and the right peak is those who are more highly expressed in the evening under a normal condition. So as you can see to the right, sleep restriction and sleep deprivation completely disturb the morning to evening regulation in between this number of genes expressed, leading to, again, to a desynchronization in between clock gene expression, and not only clock genes, but they were also metabolic genes. And after that, we also conduct another experiment. So we were interested, okay, so if we are able to sleep 4 hours, it's the same to sleep 4 hours from 11 p.m. until 3 a.m. or from 3 a.m. until 7 a.m., which is the timing of sleep restriction, because the timing is highly relevant for us, and then that was the idea. After that, we conducted intravenous glucose tolerance test, as shown here, and then a Botnia clamp with a hyperinsulinemic euglycemic clamp. And then, as you can see, both sleep restriction paradigms reduced glucose tolerance, reduced the first phase of insulin release, but was further exacerbated by losing sleep late in the night. And then here, the total amount of insulin, that is quite more accurate measure of glucose tolerance, was reduced in both sleep restriction condition, but it was more affected when sleep was lost between 11 and 3 a.m. 3 a.m. And that was also true for insulin sensitivity in those sleep conditions. So it seems to be that sleep restriction itself is highly detrimental for metabolic homeostasis in humans, but some aspects of glucose homeostasis are more modulated by missing sleep early or late during the night. We also see this effect regarding physical activity, and we used the same protocol, and we saw that late night sleep loss, that is awakening at 3 a.m., reduced more significantly the activity in the afternoon the day after, or in the evening the day after. So that is supported by additional research showing that sleep restriction, of course, will lead to a decreased physical activity and exercise, which can compromise metabolism and obesity risk. And we finally, with this protocol, we saw this food intake regulation. We tried to see if whether leptin and ghrelin were modulated by this condition. We saw that only ghrelin levels were increased in the morning after sleeping until 3 a.m. So with the physical activity condition. And regarding hunger, we saw some effects, so increased after awakening at 3 a.m., but not consistently over the day. So there is a complex consequences regarding food intake, metabolic homeostasis, and sleep regulation when we shorten our sleep period. Regarding shift work, which is a classical model of circadian disruption, where the people are awake where they should be sleeping, this is a proposal that we are looking at right now, whether shift work through several pathways can lead to obesity, which is mainly based on the circadian disruption, sleep fragmentation, and risk behaviors that are some features of this population. What we know that shift workers have altered light exposure, altered sleep, altered behaviors, and altered eating patterns. So you can see also here a list of additional aspects. First, whether these night shift workers are more exposed to higher food intake, that was a question from the previous years, and now we know that no, compared to non-shift work peers, people doing shift work, they are eating just the same amount of energy, the same amount of food compared to their peers, but they have an erratic diet even with reduced 24-hour energy intake. We took a look at this at the beginning, and we saw that there's in between shift workers here in black, they're having this more delayed eating pattern over the morning, and they have an increased eating window that is the whole time that they spend eating over the day. With Ruth here, that she is here, we saw this a complete abolished type of meals pattern over the day, and they are increasing food intake in the late time of the day. This is also consistent with others, showing that after a night shift work, people are more keen to eat high-fat food, as you can see here to the right, but not healthy food. So that is one of the additional questions that is added to this puzzle regarding shift workers. Yes, and this is results from complex experiments, which people are in the laboratory for at least 14 days, which is the constant routine protocol, and to disentangle the effect of behavior, that is eating meals, the circadian phase, either biological morning or evening, or circadian disruption here to the right. So as you can see, of course, circadian disruption increased glucose response to a meal, and also insulin response to a meal, as compared to circadian alignment. This is also true when we see the biological evening, that is eating in the evening. So for example, yesterday evening, our dinner was perfect, but it was some kind of late. So that could lead us in the long run, of course, to these metabolic alterations over time. So we are proposing to shifting the time for eating as a tool to address circadian disruption and metabolic risk in shift workers. So that's the idea, and this is a nice story from 2 years ago. There is the only paper, I think, in the field with shift workers. As you can see, they were firefighters working 24-hour shift. So they were told to reduce the time for eating to 10 hours a day. So they, as you can see, they achieved that. They compared that with a Mediterranean style dietary pattern, and they reduced some metabolic risk factors such as VLDL, particle size. They increased the overall well-being. They decreased HbA1c levels, and also some blood pressure levels. So it is feasible to induce this type of intervention, but it's highly demanding, but it is also safe. To finalize with the irregular meal timing, what do we know? We know that we are eating all over the day. We are not eating anymore breakfast, lunch, and dinner, so we are constantly eating episodically, erratic, which is here the left figure. Each point here is one eating event, and then you can see right that we are humans. We are also eating in the morning hours, so in between 12 in the evening until 4 4 a.m. And with 2 peaks, we around midday and mid-afternoon, that leads to this idea. This was in animals first. So okay, what happens when the animals are fed only during the daytime, which is their own time of the day? So as you can see, just 6 weeks after the animals fed during the daytime exclusively, they increase significantly the weight gain, even when no change in calorie intake or energy expenditure. And that leads to several type of evidence showing that idea in other animal models. And this is also to show you that when the diet is changed, for example, with a high-fat diet, the rhythms can also be impaired. So this was a nice experiment conducted under a high-fat diet paradigm, showing that the activity is not different, but of course the animals fed with a high-fat diet start to eat more when they should not, so when they should be sleeping. That's the right graph there. And then this is clock genes expression in several times of the day, in the central level and peripheral one, showing a rather dampening under a high-fat diet of this clock genes rhythm in the animals. This was also true for some specific molecules regarding lipid metabolism, specifically in highly relevant organs such as fat and the liver. So the idea now is whether we shorten the period, that will give us an opportunity to improve metabolic health. And this has been shown in animals from several years ago now. So when the animals are under a high-fat diet or a normal diet, high-fat diet here in red, the animals that are under a high-fat diet, for example, these example animals, when they are ad libitum here, or they are time restricted just to eat in this time frame, they can prevent the obesity development. And we took a look at this in an experiment, but modulating the timing of food intake. And we, as you can see, we use a low carbohydrate and a high carbohydrate diet And we also saw this our postprandial response to a same meal is rather impaired in the evening. That was not evening, was 6 p.m. as compared to the morning that was 8 a.m. even with the same meal. And this is also not news, because this is known from several decades ago, that when the food intake is in the evening, the metabolic postprandial response is impaired. But we wanted to take a look at the appetite regulation here with ghrelin above and leptin below. Under this context, we also evaluated these hunger and appetite ratings, and we saw that at the end of the experiment they were rather similar in between the morning and the evening period. So the question is this one: the hedonic regulation of food intake is also modulated by the time of day. So now we with a previous experiment that we did, we also see the homeostatic appetite in animals. It has a rhythm. So it's higher when the animal is more active here in this part. But the hedonic appetite is higher in the beginning of the diurnal period, which is our late evening. So that gives us probably a time risk period for our food intake and, of course, metabolic consequences. We evaluated this in an observational study. Then we evaluated the liking and wanting, with the liking meaning the pleasure for it overall, and the wanting the direct motivation to eat right now. And so, as you can see, when the participants were asked to eat this food in the morning, this was high caloric food, and this is low caloric, it doesn't matter. It is always higher in the evening period, which is in between 6 p.m. and 8 p.m. And that was observational, and we also saw the same thing under the experimental condition in the laboratory. So we are having a time window opportunity to intervention, probably because people with obesity are more exposed to a high risk period of the day when they could be more keen to eat more, even in the absence of hunger, which is this figure overall. The metabolic response is impaired, and the pleasure to eat is increased in the evening, even when the same food stimuli was given. So I will skip this one, just to show you that the timing of energy intake is important. Therefore, when people want to lose weight and they have 1,400 kilocalories over the day, but with a breakfast predominant compared to a dinner predominant caloric intake, the metabolic response is different. So the body weight was decreased higher in the breakfast group. Also the abdominal obesity was the same over 3 months. The glucose and insulin response was increased in the breakfast group, with lower levels of glucose and even higher of insulin, and the hunger response, interestingly, was improved by eating earlier. And that was followed by this recent paper showing that the main findings behind that is that when people with obesity want to lose weight, they will lose weight anyway, eating higher in the breakfast or eating higher in the dinner under a hypocaloric diet. But with the same energy intake or total energy expenditure, the main finding was that people eating higher dinner, they have a higher rating of hunger. This is under a field study, and this is under controlled condition in the morning. So it seems to be again that the hunger regulation was the main cause behind these metabolic improvements under a high breakfast condition. This is also the same, but restricting the time for eating in obesity and pre-diabetic subjects, showing also an improved capacity to eat. So to come to an end perspective, so we have many factors here that can disrupt circadian rhythms. Now we know that an irregular dietary patterns can be associated with both circadian disruption and metabolic disruption. And there are also additional factors because we know there's a huge variability in between individuals. So, for example, my internal time is different from yours, and that is what we call the chronotype. So this is also one thing that we want with Mariana to take a look. And so there's I would say, a good time for this circadian medicine in the context of obesity, which is one of the things that I'm very interested to. So this is some conclusions, and then I would like to give you my thanks.
Thank you very much. We have only 2 questions for reason of time.
First of all, thank you for the topic. It was very interesting. My question is related to, with all these studies, with all everything that has been done, and the topic of how time and the time of the day and the eating affects the metabolism. How does activity affect all of this? For example, mainly on people whose major activity, physical activity, is in the morning. Does this affect, for example, the metabolism, or is a variable that has not been assessed yet?
That is very interesting because, as I told you, exercise is another stimuli that we can use to entrain the system. And there are many evidence now that being active in the morning as compared to the evening is different, and there is also gender differences in between. So with men being independently of the time of day, showing metabolic benefits of exercise, but with women being more beneficial from the exercise in the morning, for example. There is no such much information on the time of day dependent effects of exercise on the circadian clocks. So that's I would say I cannot comment on that. But overall, there seems to be a differential response with some also with the type of exercise, because it seems to be that aerobic exercise should be better in the mornings, and the anaerobic or strength exercise having better metabolic effects in the evening, with evening meaning in between 6 p.m. and 9 a.m. But that is also depending on the individual, because we have this internal time. So for some part of the population, exercising in the evening could be a good thing, but for the other ones, could be better doing that in the morning. So it would also depend, and we should start to personalize somehow the interventions.
Thank you very much for the presentation. I have 2 questions. Well, I have many, but I only going to have 2. And the first one is if, right now we are with the cell phone all the time, so we receive a lot of blue light, and many times we can't sleep, and the first thing we do is use the cell phone. So there are some studies in human or animal in which both aspects are evaluated. I mean, blue light and sleep late at night, if that has any alteration in metabolic. I suppose it do, but...
Yes, so the 2 answers will be that the first part is that when you are affected by the blue light in the evening, so melatonin secretion immediately stops. Then it will depend on the duration of the stimuli, but at least it's highly reduced during the evening. So melatonin, it should be with the highest concentration even before you are sleeping. So when you are using that, let's say 2 or 3 in the morning, that will lead to a reduced or abolished rhythm of melatonin, leading to a reduced sleep consolidation. So then the sleep initiation will take longer, but the sleep quality of the resting period of sleep will be also lower. So that regarding sleep. Regarding metabolic part, we know that melatonin is high in the evening, and also in the evening we are more glucose intolerant. So there is a connection in between melatonin levels in the evening before sleep or before 12 p.m. and the level of the glucose metabolism at that time. So there is a not well known still connection in between those two. So it's very complicated mechanism, because we should not eating late when melatonin levels are high, and then whether those levels are affected by this acute bright light exposure is currently not known at the metabolic level. So that I will comment.
Thank you.
Thank you very much, Rodrigo. So we give it applause again. So our last speaker from this morning session will be Mariana Cifuentes. Dr. Cifuentes graduated as a pharmacist from the pharmacy school at the University of Chile, and she went on to obtain her PhD degree from the Department of Nutritional Sciences. In 2003, she joined the Institute of Nutrition and Food Technology, INTA, and was awarded a postdoctoral grant from the Ellison Medical Foundation. She has developed a career at INTA, becoming a full professor in 2017. She has held several academic and administrative positions at INTA since 2009. Dr. Cifuentes is chief of the Obesity and Metabolism in Geriatrics and Adults Laboratory. Her research explores the connections between obesity and cardiometabolic diseases, focusing on the role of adipose tissue and its dysfunction. So today she will be talking about obesity-related disease and adipocentric view. Thank you very much for being with us.
Thank you. I want to first thank the organizers for inviting me again to this great meeting. So I'm very happy to be here. To begin, I want to tell you a little bit about the talk. There's going to be a brief overview of adipose tissue and obesity. I'm going to talk to you about the key role of adipose tissue distribution and its dysfunction in the development of the several diseases that we generally associate with obesity. And then I'm going to talk a little bit about our studies in progress, which are very preliminary data that we are just obtaining. So first of all, the other presenters did a great job talking to you about many interesting things about obesity, but in this case I want to provide like the general view of that. In general, we all know that from a health standpoint, the obesity problem has to do with all the comorbid conditions. It's not it's a very uncomfortable disease, but it's also a very dangerous disease in terms of survival and health and all sorts of chronic diseases involved. So the WHO has defined obesity more than the BMI that we all know that it's above 30. It's an abnormal or excessive fat accumulation that presents a risk for health, right? So abnormal or excessive also involves overweight, right? So in Chile we know that 3 out of 4 people have that situation, so we are in a very big trouble that we need to create the consciousness about how serious it is. So when I talk about the adipocentric view, It means it's well illustrated in this picture where, of course, we have many reasons why people become obese. And then this obesity, and particularly the fat that is deposited in the central area of our bodies, I'm going to get into that a little bit later, causes a series of derangements all over that end up affecting all the peripheral organs, right? So I study fat, so my center is fat, creating everything else. So that's the point of view that I'm going to present today. So what happens when a person gains weight and becomes obese? It's not only that they get more fat, but the fat becomes very deregulated. So this fat changes in terms of its composition, the cellular composition, the extracellular matrix composition, everything that it secretes, everything that how it reacts to different stimuli. So it becomes fibrotic, it becomes hypoxic, and all those problems are the ones that are related to the diseases that come after that. So it's a whole overall change in the fat itself, right? So that's the paradigm that all the secretion products that start to create this vicious cycle that makes the perpetuation of the problems that end up in all the obesity associated pathologies. So starting from the fat itself. So in the main functions of adipose tissue, of course, and we all know that are the regulation of lipids, right? When we are feeding, it needs to be the safe place where we keep the fat, right? The extra energy that we have in storage, and also when in fasting conditions that we need to release that fat. So that's one of the main functions of adipose tissue, and the other one is the secretion role, and the endocrine, which is a role that is a very important one beyond energy regulation. It has to do with reproduction, with blood clotting, with a lot of activities in the body that lead to overall homeostasis. So when these two are impaired is when that we start generating the comorbidities. So these alterations are the main cause for the obesity-related diseases. And in that, talking about the lipid regulation, we have here a scheme that shows that when there's more energy intake, we have this hypertrophic effect on each adipocyte. That, of course, this hypertrophy doesn't come free, but it comes with a lot of changes. And this adipose tissue becomes insulin resistant, becomes inflamed, and all these effects create a situation in where the adipose tissue is not longer able to safely keep this fat, but the fat starts spilling to the rest of the body, right? So in that sense, the spillover causes this endosystemic inflammation also because of the changes in the balance between inflammatory and pro-inflammatory and anti-inflammatory secretion products, create the lipid accumulation and inflammation in distant organs, and in this case in liver and muscle, which are very important metabolic organs, which then also create the overall the insulin resistant, and leading to diabetes and hypertension and all the cardiovascular problems that we relate to obesity. And in terms, of course, of the secretory products of adipose tissue, we have many factors that will affect different organs, generating all the diseases that we know. So these I wanted to show you here how the alterations of the main functions generate these diseases. Also, adipose tissue plasticity is very important. Adipogenesis is not a function of the adipose tissue, but it's also a way that adipose tissue has to adapt to the different situations, mainly overeating, of course, and it has to deal, as I said before, with the extra fat, right? So in general, in healthy conditions, we have here that there's a positive energy balance, and adipocytes react by increasing their number and increasing their size. And there has to be a balance so that there's healthy adipose tissue expansion. It has to deal with the extra intake. And then we have, like I said before, this safe lipid storage, which is called, and I'm going to get into more into that, like the metabolically healthy obesity, right? So we have this situation where there is obesity, but it's managed. Okay, so there's no insulin resistance and no hypertension, and we are okay in terms of this adipose tissue doing the job, right? But what happens when there's defective adipogenesis? We have so that we cannot increase the fat deposition through new adipocytes, but the existing adipocytes become bigger, right? Become hypertrophic, and as I said before, that means that they become altered in all their functions. So what we have here, when there's an inhibition of healthy adipogenesis, we have adipose tissue dysfunction, inflammation, everything I said before, and then we have what is called the metabolically unhealthy obesity. So adipogenesis is a very important way that adipose tissue has to maintain its homeostasis. And here another picture to reinforce the idea. So pathologic expansion means mainly via hypertrophy. The adipose tissue becomes inflamed, altering the secretion and everything I said before. But when we are able to do it mainly by hyperplasia, we have an adipose tissue that is well vascularized and not hypoxic, so we have more time to deal with this. Of course, eventually it will become insulin resistance and everything, but we'll have here a window where we can still maintain the metabolic health. So in these terms, I mentioned before that central fat was the one more dangerous, and here is a picture depicting exactly that. Visceral obesity is the one that is more related to the pathologic effects, right? So in general terms, subcutaneous obesity, especially gluteofemoral depots of adipose tissue, are actually more protective. So in general, what we worry the most in clinical settings is when obesity is the apple-shaped type of obesity as opposed to the pear-shaped, where fat is mainly, above or below the waistline. And that is metabolically associated with a lot of differences, and also in terms of molecular effects, the different fat depots are actually very, very different. And I think in a few years they will be called different tissues because they are so different in everything. So that leads to the concept that I mentioned before and that we have been very interested in the lab, of this metabolically healthy and unhealthy obesity. We can talk a little bit more about that in the question section, but of course it has a lot of controversy. But in general terms, if we only think about obesity, we see that these two phenotypes are very, very different. And as I said before, it has to do with adipocyte size, it has to be with the secretory products, it has to be with fat distribution. And as you see here, in healthy obesity, fat is restricted to fat tissues, whereas here fat is also in the liver, in the muscle, of course, disrupting their function. So I'm going to go back a little bit later on with that concept, but I just wanted to stay a bit talking about the interaction between disrupted fat and the other organs and how that leads to obesity. To the diseases of obesity. And in general terms, the mechanisms are very similar, because we have here that in general, fat in obesity leads to the secretion of many factors that will, through the bloodstream, reach, muscle, liver, and other organs, and over there generate all sorts of disruptions, leading to inflammation there and insulin resistance there. And in liver specifically, it will lead to fatty liver, which we all know that it induces a lot of diseases. And in terms of cardiovascular disease, here also liver helps after receiving all the signals from specifically central fat, also leads to the production of many clotting factors and alterations that will lead to atherosclerosis. And also here, fat through the systemic circulation will reach the heart and also create the inflammation there. And in addition, there's also local depots of fat within the heart. One of them is the epicardial fat, epicardial adipose tissue, that also will endure all these changes that will affect directly the heart muscle, generating all sorts of problems there in obesity. So the local depots are very important in terms of cardiovascular and all diseases too. And other diseases that are very related to obesity, for example, cancer. Dr. Quest tomorrow will give a talk about that. So this is like the preview for that. That there has been established that at least 13 types of cancers are really related to obesity, and he's going to talk more about that tomorrow. And the other talk tomorrow that I'm advertising today is that of Dr. Daosawa, who is going to talk about senescence. So because obesity, as you see here in this picture, has many common features with aging. So what's considered that obesity is an accelerated aging model. So he's going to also cover that, and it's very interesting because they have so much in common. And to finish, I want to show you a little bit about our ongoing studies. And as I said, we have been very focused on the two obesity phenotypes, the healthy and the unhealthy. And we are doing an intervention of intermittent fasting. Rodrigo already mentioned this is time-restricted feeding. Remember those great studies that the rats, the mice, didn't get all the diseases if they were fed on a very restricted time. So this is what we are trying to do in these humans with obesity, to see whether there's an effect in the differential effect in the metabolically healthy and the metabolically unhealthy. So our protocol is a window where they eat for 8 hours, and the 16 hours are complete fasting, except for water, of course, and for 16 weeks. So we've been working here, Gabi, Nati, and Lily, we are present here, and I really thank the organizers that they could come to this meeting, so you can go to their posters and see more of that. But in general terms, so one of the things that we do, we took the blood from these volunteers, and we isolated peripheral blood mononuclear cells to expose them, among other things, but we exposed them to vehicle or LPS. This is a pro-inflammatory stimulus, and we look for many things, some of which I'm going to show now, and the others you can see in the posters. So very interestingly, we see that this time-restricted eating caused weight changes, weight loss, and weight circumference loss in these subjects. And this is quite impressive. I'm dividing this in unhealthy and healthy obese subjects, and the effect was the same in both. But it's very interesting because for a protocol that did not restrict energy at all, there was no calorie counting, they could eat whatever they wanted, but they just had to eat within the 8 hours of their window. So this is very impressive, and compared to other clinical trials with weight loss, caloric restriction and everything, this 2% approximately weight loss and this 2.5% of waist circumference is quite interesting. Remember, this is no calorie counting here and no restriction whatsoever. So we were very happy to see that this really works. And one of the results here is that this is gene expression in the PBMCs, these peripheral blood mononuclear cells. So this is the expression of pro-inflammatory markers, TNF-alpha, IL-6, and IL-1 beta. And we see here the effect is the same, so I'm going to use one as an example. So in metabolically unhealthy subjects, there's a larger inflammatory response to this, whereas in the metabolically healthy, actually there is no significant response to this inflammatory stimulus. And in terms of the secretion to the medium of these same factors, we see also that the change, this is in black, it's the unhealthy, in blue is the healthy subjects, and what we see here is that there is a difference in the change in the response to LPS. So there's a larger response in the unhealthy. It's consistent with what we just saw, but this is the secretion of the cytokines to the medium, right? So this is what happens at baseline. And interestingly, What happens after the 4 months of restriction, there is no difference in the reaction between the unhealthy and the healthy subjects. They are the same. There's no difference, as you can see here in the p-values, in the reaction to the inflammatory stimulus. And it's very intriguing and it's very interesting because actually the response, if you see the percentage numbers, is much larger. It's much larger than the response at baseline. So this is quite interesting, and we're trying to interpret these results. But something is going on, and again, as I said, this is a very preliminary data. There's an avalanche of clinical data coming to us in a few weeks, so we are going to have a lot of work. So this is the first things we are able to see, and you can see again the posters. There's something about the clock, gene regulation too, and I'm going to leave there to explain it. And with that, I want to thank you for your attention and all the people that have been involved in this. Thank you very much.
Thank you very much, Dr. Cifuentes. Do we have some questions? Okay.
Thank you very much. I'm curious, the time restriction that you used in your last experiment that you showed was in the morning or in the afternoon?
Right, that's a very interesting question. It was free. The subjects had to pick the timing, and that when I designed the study, I didn't really consider the relevance of the timing. And it's definitely, I mean, they are diurnal, that's one thing. I mean, but some of them start eating at 1 in the afternoon, and that's not as desirable. In the best scenarios, and in the future studies that I'm going to do, I will enhance the concept of the early time restricted intake, because it's been proven that, as Rodrigo just mentioned, that early is when you are supposed to be eating rather than later. Because of the whole metabolic reaction the different metabolic reaction to early eating versus later eating.
I agree that the morning will be better, but we are late people. Most of us, we have our social gathering at night. So
exactly.
I will be interested in to know
Absolutely, so very interesting. Yes.
The two options that is best for him. Thank
you.
Mm-hmm.
Thank you.
Further questions?
Hello. First of all, thanks for the presentation. Was very good. But about the fasting, they have that 8-hour lab where they could eat whatever they want. It was like that, or not?
If they could eat whatever they want?
Yeah.
Yes, it was actually, there were no restrictions. They were asked only to eat within the 8 hours that they decided when those 8 hours were, and they had no restriction.
Yeah, and obviously a lot of them lost weight, but what about exercise? Do you have any data about exercise they've done, or?
We have the data of their their habitual exercise, but there was no intervention. I mean, they had to do their life, and the purpose of that was to intervene the least possible, to see whether in real life this would be feasible or this would be effective. So I tried to design a study where the minimum of effort would have to be from the subjects, so they wouldn't change a lot their lifestyle other than the restricting their hours of eating.
Okay, thank you.
Sure.
Any other question?
Thank you. It's just a very simple question that I have from the previous talk and this one also, very nicely done. You're looking at obesity, and I know that the literature is not really showing much data in the intermediate moment, the overweight patients.
Yeah.
So what is your view about overweight and the worries that we have to have for the future?
Right, that's an excellent question, thank you, because I don't know whether to say unfortunately or fortunately at this point, but in the recruitment, it was supposed to be only people with obesity, but we have an interesting number of people with overweight. So we can see some things that are very interesting in terms of their reaction to this. But this is a work in progress, so I don't, but you're absolutely right that of course there is a risk there, and in terms of the phenotypes, there's a lot of information that even lean people with central fat, or the one that you know, like the liquid paper phenotype, they are more at risk than people with obesity that is peripheral obesity. So definitely, I think there's a continuum. I think that the IMC of 30 is very, how to say, not so well based on real life. It's just very epidemiological. It only works for large amounts of people. But I think that it's a continuum. So I wouldn't differentiate it much. And maybe, fortunately, I have data, so I can answer you in a few months, your question.
Thank you very much, Mariana. Very interesting work, and we give her an applause again. And thank you to all the speakers this morning. It was a very interesting session. So we have now 1 hour for lunch, and then we continue with the poster session during the afternoon. Thank you very much to everyone. Okay.
Vamos a, hay un lunch, un almuerzo.